I will, kind of the second half, I will highlight three projects that I have been involved with that have leveraged sort of the clinical trial infrastructure, and also speak to sort of, I think, some of the new promises that I'm involved with in the field here at the University of Minnesota. So thank you all for participating. So the first two slides, I am indebted to Dr. Brandon Diesner, a postdoc in Logan Spector's lab that I work very closely with, and just wanted to really highlight one of the sort of key challenges of doing research, and especially trying to do translational research in sarcoma, is that this is a very rare group of malignant tumors. And also, it is a very broad group. So it involves multiple different tumor types. And depending if you're a lumper or a splitter, it can be anywhere of up to, close to 50 to 100 different tumor types, or you can lump them into broad categories. But it makes for significant challenges in studying sarcoma. Now, the other thing is that the types of sarcomas vary by age. And so in the pediatric population, this is data that Brenda put together from the SEER registry, there is sort of this peak in young children of rhabdomyosarcoma. In sort of the adolescent young adult, we are really seeing predominantly osteosarcoma and Ewing sarcoma, and synovial sarcoma sort of spans the lifespan, and is the most common non-rhabdo soft tissue sarcoma seen in the pediatric age population. However, if you look at sarcomas in the adult space, it is a very different tumor type. And with leiomyosarcoma, a muscle tumor, being the most common, followed by gastrointestinal stromal cell tumor as the number 2. But what you'll also notice here is this is incidence, the numbers per 100,000. So you're looking at here, and if you look at the different types in children, this is less than 1 per 100,000. And on the adult side, you're running in the 1 to 3 patients per 100,000 individuals, much less than the 1 in 8 women who have breast cancer, which highlights some of the real challenges of doing sarcoma research. So we at the University of Minnesota, many years ago, and really with the impetus of the Karen Wyckoff-Rainin Sarcoma initiative, really mobilized around developing a true sarcoma program. And our program really recognized that unless we focused and worked together, we were not going to make strides in these rare tumors. And so cornerstone to our mission was that this was interdisciplinary, meaning expertise in many areas. It was collaborative, and that we would understand the etiology or the causes of sarcomas to try to better develop therapies. Because unless we understand the biology, we can't really understand how to develop new therapies. So we really focused our program around the strengths here and the expertise. So really the leveraging our expertise in gene environment interactions, genetics, and epidemiology, the associations within populations. We have a very strong comparative oncology program, and I'll highlight that in a couple of my examples, because I work very, very closely with Dr. Modiano in comparative oncology. We have strengths in cell-based therapies and biomarkers, and kind of my main role is really that translational piece, moving the development and the information to the clinical interface. And so the sarcoma program currently has over 40 members in 9 departments and divisions at the university. And this is this real multidisciplinary approach. We really need expertise in all of these areas. And these are really departments and divisions that house both basic scientists and translational and clinical folks. But we need all of this to actually make strides in this group of diseases. And so when we talk about the goals, and I really talk about translational research, it's bench to bedside and back, because we have to look at the basic science side of things, the basic scientists. We have to take those findings to the patient, to the bedside, but then we have to take those learnings from the clinic and take them back to the basic scientists. So it's really this ongoing loop that all of us have to collaborate together to move these findings and discoveries from the lab to the clinic to benefit our patients. So I typically refer to it as bench to bedside and back. One of the key principles of treatment, and this you'll get to sort of, David was referring to in my roles. Not sure what happened there. We will go back to sharing. Not sure where it went. Can you see it?
Yeah, that looks
okay.
good.
Fantastic. Not sure what happened. So one of the things, the real principles of treatment, is to get to the clinical trial, to get to moving these discoveries to the patient. And in pediatrics, we really have a culture of clinical trial enrollment, and about approximately 80% of pediatric patients are enrolled in a clinical trial at some point in their cancer journey. This is compared to actually less than 5% of adult oncology patients. And at the University of Minnesota Masonic Cancer Center, actually, we are higher than this national average. We're running at about 14% of adult oncology patients. The real hallmarks, historically, two principles of treatment for sarcoma have been surgery, chemotherapy, and radiation. And we're now moving into the space of newer agents and immunotherapy. One of the things we know, and one of the things that's so important about getting to the clinical trial piece, is that patients who enroll on clinical trials, and this is data not from sarcoma, but it's actually from Britain, where they summarize people who enrolled with Hodgkin's lymphoma, lung cancer, and leukemia. And what you can see is this is overall survival in years from follow-up. And the people who were enrolled on clinical trials, which is the blue line, had an statistically increased survival compared to patients in the red who did not enroll on clinical trials. So it actually improves overall survival because of coordinated and standardized care. So there is a real reason beyond just the drugs to get on a clinical trial. So sarcoma research has several challenges. It's a relative low incidence, which I showed you very early on from Brandon's data. It mandates multi-center, multidisciplinary clinical trials and collaborations. We're even now in the molecular age, we're developing smaller and smaller subgroups as we learn more and more about the biology. But we have to work together to improve outcomes. And a lot of this has been really driven, as David said, through support from the National Cancer Institute and Cooperative Group Program. And I have been very involved in this with regards to the Children's Oncology Group. And so the Children's Oncology Group is really designed to conduct large clinical trials and small clinical trials, but it really serves also as a repository for centrally collecting biology samples, and I'll refer to that in the third example that I'm going to share with you, to translate new findings so that we have biological samples to work with for these very rare tumors. We really need to look at causes and associations and also improve quality of life and survivorship. So the Children's Oncology Group is made up of over 220 institutions that is actually essentially becoming a worldwide organization, North America and Canada, Australia, New Zealand, and we are starting to get into Asia as well. What my main role within the Children's Oncology Group is I actually am the principal investigator for a separate UM1 grant for the Pediatric Early Phase Clinical Trials Network, which is 21 sites that are selected from within the Children's Oncology Group. This is their distribution to do early phase trials. So to bring those early discoveries to the clinic, but to do it at the national level. So one of the goals that we as a group have had, and as I said, we've sort of focused, is we early on developed a focus in osteosarcoma. And this has really been driven by work from David's lab, Brandon Moriarty's lab, and Beau Webber's lab. And really, we haven't made any strides in over 40 years, despite surgery and chemotherapy with lots of side effects. So Brandon discovered that it was trying to identify genes associated with drivers of osteosarcoma. And we know one of the big drivers is P53, which is incredibly difficult to target. But what he did discover was a gene SEMA4D. And interestingly, there was a drug called VX15 that actually targeted SEMA4D. And so what the SEMA4D story really is that Brandon was able to focus and define that SEMA4D was a strong candidate and is essentially expressed in almost all human osteosarcomas and at high levels, and that there is also a really strong interaction with the tumor microenvironment, and there is modulation of the immune system by modulation of SEMA4D. What was really key to driving this translation was that VX15 was a humanized murine IgG4 monoclonal antibody that bound to SEMA4D that was available through a company called Vaccinex that was very interested in moving this into the clinic for osteosarcoma. So Dr. Emily Greengard in our group led a national trial through the Children's Oncology Group Pediatric Early Phase Clinical Trials Network that I described. 4 patients, first pediatric patients with solid tumors to define the dose, but importantly, we had a cohort of patients targeting for osteosarcoma. We defined the recommended dose given every 2 weeks. We studied pharmacokinetics, looked for anti-drug antibodies since this was a murine-based drug, looked at pharmacodynamics. Very importantly, one of the things that was correlated in adults with potential efficacy was looking at T lymphocyte saturation in response to VX15 as a sort of surrogate biomarker for the drug. And also, we required tumor submission to look at SEMA4D expression. And another key marker that Brandon has defined in his lab is plexin B1. So we enrolled 26 patients, and of those, 8 patients who were evaluable in the first part, and part B is the osteosarcoma cohort. Unfortunately, we did not see any patients who had a true what's called RECIST response, but that is a very difficult marker in osteosarcoma to look at. That means shrinkage of the tumor. But we did have 2 patients that had prolonged stable disease, but that did not meet the bar for defining an efficacious dose, which would have been 4 patients. But what is interesting, and we are hopeful that we can learn something from especially the patient that stayed on for considerable number of months on drug. So through this national effort coming out of the Moriarty lab, the drug was very well tolerated. We have a dose that we know is safe. We're looking at all of those correlative studies and really taking that next step back to the bench to say, can we learn something from some of these tumor markers, and then look at combination strategies to try to then go back to the clinic. So this is the real paradigm of bench to bedside and back. Another agent that I am now very involved with in developing for sarcoma that has been a real collaborative effort across our whole program that has at many levels had support from Rain and Sarcoma is utilizing what's called a bispecific angiotoxin, EBAT, and this has involved several collaborators here. The drug was developed by Dr. Dan Valera here at the Cancer Center. And it's bispecific because it targets two receptors on cells in the tumor microenvironment and on the tumor. The epidermal growth factor receptor and the urokinase plasminogen activator receptor. These bind and are attached, well, these bind the microenvironment, I'll show you on the next slide, but they're attached to this pseudomonal endotoxin, which is really sort of the cargo, if you will, that is carried to the cells when it binds to these two receptors and causes injury to that microenvironment and cell death. So this is from a lovely paper by Antonella Borgatti in comparative oncology, demonstrating how this works with the binding of both to EGFR and to UPAR. And then what happens is that starts to disrupt both the microenvironment from a vascular point of view, an anti-angiogenic point of view, but also there are effects on the immune system. So it affects the tumor cell, it affects the stromal microenvironment, making it inhospitable to the tumor cell, disrupting blood supply and immune microenvironment. And it allows the cells of particularly macrophages to counteract some of the immunosuppressive microenvironment effects that tumors are known to affect. So multiple models through Dr. Modiano's lab and Dr. Borgatti have really discovered that there are anti-tumor effects in osteosarcoma cells. So this is using the EBAT is killing osteosarcoma cells in culture. A former fellow of ours, Christy Pelbeam, in collaboration with Dr. Valera and Dr. Mike Vernieris, actually studied mice with rhabdomyosarcoma and Ewing sarcoma. And you can see here in the mice that received the higher doses of the EBAT, there was decreased tumor growth from rhabdomyosarcoma and decreased tumor growth in Ewing sarcoma. And Dr. Borgatti has treated dogs with hemangiosarcoma, a vascular sarcoma that occurs in dogs in the spleen, where they're treated with a splenectomy and adriamycin, which is this control arm here. And when you add EBAT to that regimen, there is improvement in progression-free survival and delayed recurrence. So 3 different models representing cells, mice, and dogs, suggesting effects in sarcomas. So our goal for the EBAT drug development program is to complete the characterization of the mechanism of action, because we really want to understand this interaction with the tumor microenvironment. We are in the process of submitting an IND to do the first in human trial with a sarcoma expansion. And we are really hopeful to submit this by the end of 2020 to begin the trial, hopefully in 2021. The third example is a collaboration again with Dr. Modiano, myself, and Dr. Spector, really trying to develop a biomarker for predicting and stratifying patients with osteosarcoma for risk of recurrence, so that we can ultimately help guide therapy for patients with the highest chance of poor outcome, but also decrease therapy for patients who may not require as an aggressive an approach. So the exosomes, this is a hot area, are really sort of small vesicles that are secreted from cells as they divide and contain microRNAs that can be considered tumor signatures that can be detected in the circulation or in the blood. And what Dr. Modiano found in mice was that there was a particular signature, stage 4 is with metastatic disease, that correlated with progressive disease, particularly a marker CD63. So he further went on in their group to define this signature, and that it was really unique, this CD63, to the exosome lysate, and was able to be characterized as associated with the tumor. So what the real goal of this project is essentially to isolate and extract these exosomes from mice, and also, and then our goal is to sequence the RNA for the signatures, and then to really validate it between cases in dogs and in humans, and so that we can really utilize all three models for this predictive biomarker. So they have isolated with looking at, again, at between controls and at week 8 in osteosarcoma in mice, a very clear signature that can be utilized and in a replicated fashion. And so using this machine learning with this exact signature, we were able to predict in dogs, in a set of samples from dogs who had osteosarcoma and who did not have osteosarcoma, that this biomarker was predictive of the potential for recurrence. So our goal for this project is to now utilize samples from the Children's Oncology Group osteosarcoma studies, so accessing that central repository of tumors, and then to match or exceed the prediction that we achieved within the canine, and hopefully be able to detect minimal residual disease as a predictor of response, essentially looking at this as a liquid biopsy or as a biomarker for responsiveness that maybe someday we'll be able to tailor therapy. So we're very hopeful that this ultimately we will be able to actually develop this as a bona fide biomarker for osteosarcoma. So really, the hope for our future takes a team approach. We need a collaborative research environment, which we have, thankfully, as I hopefully have demonstrated to all of you. It takes a village, truly. We need people with population experience. We need lab scientists in the preclinical space, and we need clinical expertise to move it from bench to bedside to back. We have to be able to improve the lives of our survivors through these efforts. And we are really indebted to the patients and families who participate in research, because it is the only way that we have actually been able to do these trials and actually to even think of doing sort of a large scale study of the biomarker for the samples that have been collected. So one of the things I really wanted to emphasize is how critical the Karen-Wyckoff-Rainin sarcoma research and support has been to this effort. To highlight, since 2004, 22 research grants have been awarded to the University of Minnesota, totaling just shy of 1.6 million. This does not include recent grants. So the recent grants, the giving is now over that 2 million mark. These grants have resulted in 58 peer-reviewed manuscripts and 25 additional grants, thus far totaling 13.5 million. We do have 6 ongoing projects funded the last 2 years, and several of these you will hear about today, and really provide that hope and promise for the future. In 2016, Dr. Larry Seymour honored the memory of his granddaughter, who unfortunately lost her battle with osteosarcoma in forming the Hallie Ann Brown Educational Initiative. And it is really thanks to Dr. Seymour's transformative gift that we mobilized into developing this incredible symposium, but also really developed the research funding task force that was his vision for expanding the funding to include the Mayo Clinic and Children's Hospitals and Clinics of Minnesota. And you will hear reports today from both of them as well. And so we are indebted to Dr. Seymour, but most importantly, highlight Hallie Ann's memory. So in closing, I just want to acknowledge the tour de force team that I have the privilege of being a small part of here at the University of Minnesota. My colleagues in leadership at the Children's Oncology Group. I am privileged to have several funding sources and most importantly, thank patients, families, and who participate in research, and the dedicated pediatric oncology community who makes this all possible. So with that, I will close, and I apologize for that technical glitch in the middle. Not sure, my computer.
Thanks so much, Brenda. I'm now going to hand over the Master of Ceremonies duties to Logan Spector, who will take over for the next part of the meeting.
Thanks, David, and welcome everybody. And thank you, Brenda, too, for an excellent overview of sarcoma research, both nationally, internationally, and at the university.
Yep, I'm here, and looks like I have my share screen button.
Great. So it is my pleasure to introduce Beau Webber, who is assistant professor in the Division of Hemonc in the Department of Pediatrics, and you know, you'll see a theme throughout his presentation, as well as in some of the student posters, that the university is developing induced pluripotent stem cells as a way to model sarcomas. And I'm particularly enthused about this project because it's a chance for me to work with Beau. I have zero skills in handling cells, but it's a nice collaboration where he's doing the cellular work and I'm helping with the population genomics. And so Beau, take it away.
Yeah, sure. So you guys see my presentation screen?
Yes, we do.
Okay, excellent.
Beau, you may want to put it into slideshow mode.
Oh, yep, sorry.
Okay, let's see. How's this?
Perfect.
All right, great. Yeah, and as I know next to nothing about population genetics and epidemiology, it's been likewise a great collaboration and you know, really kind of opened up new ways of thinking about things, and I've really enjoyed it so far. So the original title of my talk was more focused around some of our work utilizing ancestry-informed genetics, but our work in this space on Ewing sarcoma has branched out since that initial proposal and the start of the project through Rain and Sarcoma. And so I've broadened the title here with this talk to simply describe our work on unraveling really the etiology of Ewing sarcoma, as Logan said, using pluripotent stem cell technology. So Ewing sarcoma is a cancer that originates from bone or the soft tissue surrounding bones. Histologically, it's characterized as a uniformly undifferentiated characteristic of small round blue cells. It's notably very genetically quiet, aside from the driving EWS ETS fusions. And it's also very unclear still, the developmental cell of origin. So although it's suspected to arise from mesenchymal tissues, as I'll just talk about it shortly, the real sort of puzzle of within that group of mesenchymal cells, where this originates is still unclear. And so those are really the big questions that we're seeking to answer. Namely, how do the developmental and genomic context influence the initiation of Ewing sarcoma? And so on the genetic level, it's well characterized that the vast majority of ES patients have a translocation between EWS and ETS family of proteins. Specifically, the most common being a translocation between EWSR1 and FLY1. And so the fusion protein created by this links the activating domain of EWSR1 to the DNA binding domain of FLY1. And this fusion protein is now known to preferentially bind to GGAA microsatellite repeat elements throughout the genome. And what this results in is the alteration of transcription of many genes, essentially global misregulation, both activation of genes and inhibition of genes. And interestingly, when you express this protein in cells in culture, it's very toxic to the vast majority of cells, and it's not well tolerated. So that's one of the big questions that we'd like to answer is how does this protein enact a program in the absence of other associated mutations, because Ewing sarcoma is one of, as I mentioned, one of the most genomically quiet sarcomas aside from the fusion protein. So how this Ewing program is established still remains largely undefined. The second layer of complexity with Ewing sarcoma is the developmental origin, so the cell type from which it arises. And as I mentioned, it's suspected to arise from a mesenchymal origin. This is just a kind of a broad definition of mesenchyme, kind of trying to illustrate how it's kind of a poorly, loosely organized sort of collection of different connective tissue types. And this is really kind of based on the anatomic location from where Ewing sarcoma arises, its proximity to bone, and its involvement in, for instance, cartilage, bone, and the soft tissue surrounding bones. And so really one of the cell types that's been focused on are these mesenchymal stem or stromal cells, which can be isolated from bone marrow and adipose tissue and are capable of forming multiple lineages, primarily bone, cartilage, and fat. But this population is pretty vaguely defined still. And the evidence further supporting MSCs as being a potential origin of Ewing sarcoma has been shown in a few studies. For instance, by knocking down EWS-FLY in ES cell lines, it shifts gene expression towards that of primary MSCs. Similarly, several studies have demonstrated to a certain degree that EWS-FLY is tolerated in MSCs. And that's kind of based on your definition of tolerance, but suffice it to say that MSCs have been shown to propagate, for long-term expressing EWS-FLY. But at the same time, there's also a fairly robust amount of evidence that Ewing sarcoma can originate from a neuroectodermal origin. And so MSCs are primarily derived from mesoderm, which is one of the 3 germ layers, whereas neuroectoderm or neural crest cells derive from ectoderm. And so a lot of this is based on the observation that ES tumors often exhibit characteristics of neuroectoderm. It's also been shown that induction of EWS-FLY in, like, for instance, NIH323 fibroblasts induces a neuroectodermal differentiation pattern. It's been found that if you overexpress EWS-FLY in other sarcoma lines, it drives a neuroectodermal gene signature. And once again, you can always find sort of some evidence that of a neuroectoderm or neurocrest lineage has been shown to tolerate EWS-FLY fusion. But the complication of this is that MSCs themselves can actually arise through ectoderm from neural crest cells. And so within this developmental spectrum, it's really unclear exactly where the cell type or which lineage is the one that's able to be transformed by EWS-FLY and is really the sort of primary lineage that's formed in vivo, that gives rise to Ewing sarcoma in vivo. And one of the limitations of the studies that have been conducted to date is that they've all been done in cells that are under the influence of EWS-FLY expression. And so, as I mentioned, EWS-FLY alters many genes, and so you can imagine how, enforcing this program and looking at the cells under the influence of this program could impact the observations and your results. And so this was really one of the early things that Logan and I teamed up on, which is based on this work here. This is a really good example, but there are others as well, is that the chromatin organization can be mapped to mutation or genome variants. And when those two things align, so open chromatin, when it aligns with either variants or mutations, is very predictive of the cell type of origin. So like for instance, in melanoma, the chromatin profile of skin aligns most closely with the mutation profile that you see in melanoma. And so our hypothesis was, could we take the genome variant data that Logan had generated in his lab and overlay it to open chromatin profiles in developmental intermediates to identify a putative ES cell type of origin? And so you can imagine how this might work. Just showing here what, a depiction of a closed chromatin, open chromatin, and then 3 hypothetical cell types: blood on the left, which is unrelated to Ewing sarcoma, neural crest cells, and then some variation of neural crest, we'll call it the putative cell of origin. And so we have this genome, we have this variant data set, and what we want to hypothesize is that in these lineages or intermediates, we would see these variants overlay primarily with closed chromatin in unrelated tissue types or unrelated lineages. But as you got closer and eventually settled in on the cell type that most closely replicates the cell of origin, you would see these variants predominantly fall within open chromatin. And so the question became, how can we actually access these developmental intermediates? It's very challenging to do this from human beings, of course. And so that's where we turn to the induced pluripotent stem cells. So for those of you who have not heard of Ips cells, they're essentially a pluripotent cell type similar to an embryonic stem cell that can be created by reprogramming somatic cells. So this was shown by Shinya Yamanaka. You add the Yamanaka 4 transcription factors and can reprogram fibroblasts or blood to these Ips cells. And these cells can be propagated long-term in vitro and can differentiate into virtually all somatic tissues, including, in our case, the tissues that we were interested in, namely mesoderm and neuroectoderm-derived progenitors and mesenchymal cells. And so our goal really for this project is to utilize this system to develop these developmental intermediates and establish open chromatin profiles at different stages of development, and then compare these profiles to the ES-GWAS data set that Logan has generated to identify these cell lineages with a very high degree of overlap. And so we set out first to demonstrate that we could actually generate relevant lineages. And so here you see on the top left is an Ips cell colony. And we can trigger this to go down the route of mesoderm. Won't get into the details just in the interest of time. But once we've induced mesoderm, we can subsequently specify mesenchymal differentiation and generate these cells that look morphologically just like bone marrow MSCs. They express all of the surface antigens that MSCs you'd expect from MSCs. This is just a comparison to bone marrow MSC. And they're able to form osteoblasts, chondrocytes, and adipocytes. And so this is actually something that we had worked out prior, but one of the things we hadn't worked out was how to go through the neural crest lineage to the MSCs. And so this is actually where we are collaborating with David Largaspada's lab and Garrett Draper, who's studying MPNST using Ips cells. And so he's really helped kind of establish this protocol in our group, being able to generate neural crest cells through neuroectoderm. And these cells, again, express the surface markers that you would expect for neural crest. They lose the expression of the pluripotent factors and upregulate neural crest factors. And these neural crest cells can then be treated essentially in the same way as mesodermal progenitors to drive mesenchymal specification to generate these MSCs. And so that's essentially right now where the project's at. So Kelsey Becklin in our lab is leading this, and she has, despite the COVID hibernation, been able to generate a large number of these intermediate developmental stages from multiple donor, Ips cell donor lines. And so far we have iPS cells, mesodermal progenitors, 3, I'll just call them flavors of mesodermal MSCs because there are different sort of variations of these, but suffice it to say we've submitted three of these, neurocrest cells, neurocrest MSCs, and then we also threw in a number of primary different MSC tissues that would be more sort of the terminally differentiated, just as an added sort of measure to see where our cells fall in the line, because presumably some of these, as derived from iPS cells, are going to be an earlier, more sort of like embryonic or fetal stage of these of these cells. And so the next step, and what is actually occurring right now, is that these cells have all been treated using a protocol called ATAC-seq, which uses a TN5 transposome to land an open chromatin. It brings in an NGS adapter, and then following NGS, you can map these reads to regions of open chromatin. And then once that data rolls in here in the next month or so, it'll go off to Logan's group to start doing the alignment. And so we're very excited to see where that goes. So just switching gears now to the genetic side. One of the things we've really been interested in, especially Logan, he's the one who really stares at this kind of data all the time and thinks about it, is this really stark disparity in the incidence of ES among ethnicity. And so Europeans have the highest incidence of ES, with Latino children having about 2/3 that of European or white children. However, when you look at all bone sarcomas and then compare that to Ewing sarcoma, what you see is this incredibly stark disparity in adolescents of African ancestry, which is about 10-fold less than that of white children, and essentially no occurrence in any children above the age of 15. And so presumably there is a genetic link that causes this, and that's what we want to try to investigate, or our hypothesis was that we could investigate this using iPS cells. And so one of the things that I briefly mentioned at the beginning of the talk is that there's this growing evidence, and it's now quite well established, that this EWS fly fusion protein binds these GGAA microsatellite sequences. And the length of these sequences dictates the binding affinity of EWS fly. What's now being more appreciated is that some of these microsatellite differences are actually variable based on ethnicity, specifically between European and those of European and African ancestry. And in fact, a recent publication found that, so that in this study, they looked at the genome-wide chromatin binding by EWS fly. And what they found is that 36 of the bound GGA repeats have been shown to be statistically significant difference in lengths between European and those of African ancestry. So it really points to this concept that perhaps in so-called permissive genome or a European genome, the composition and lengths of these GGA repeats throughout the genome are set up in a way that sort of creates a fertile ground for this Ewing sarcoma transcriptional program to establish itself. And so hypothetically, if one was able to induce EWS fly in cells spanning European and African or permissive or impermissive genomes, you could potentially use this to identify specific transcriptional or epigenetic changes or loci that are the key to driving transformation in ES. And as I had mentioned, there are hundreds of genes, thousands of genes that are misregulated, but understanding the very few of those, the subset of those that are specific to driving the transformation process, is still an active area of investigation. And so we thought that this approach might be a novel way to try to address this. And so we started looking at iPS cells, of course, and when we started looking at these large stem cell banks, such as Y-cell, we were able to find collections of iPS cell lines that span, that have defined ancestry, and particularly this gene star set at Y-cell contains a diverse set of iPS cell lines that span from essentially from 0 to nearly 100% African ancestry, and also have, you know, within the same collection, European ancestry as well. And so with our initial funding from Rain and Sarcoma grant, we actually went out and were able to obtain a collection of these lines. We started out with 8 initially, and that is growing now. As you can see, we were able to obtain several with 100% European or African ancestry, and then some, you know, within the span of 50% to 100%. And recently, we received an AARP grant to expand this dramatically. And so these are the lines that we are going to be acquiring over the next couple months to really broaden and expand the number of lines that we have. And so you'll see here, as I'll describe in the next slides, we have sort of a 2-phase approach to how we're going to go about this. As you can see, this is a very large number of iPS cell lines, and we're actually going to be working with James Dutton in the Stem Cell Institute and the Center for Automation there to use their sort of robotics to be able to do some high throughput evaluation of the response to Ewing EWS fly in these cell types. And so I just love this acronym. So the main goal of this, what we want to be able to do is, and this is Logan's came up with this, I'll give him credit, but is identify what we call ancestry-linked differential Ewing response loci, or ALDR loci. And so to do this, the plan is to take essentially these iPS cells of European, African, and in this case, Amarin ancestry, differentiate them to the neural crest and the mesodermal mesenchymal cells, and then introduce EWS fly fusion protein as a synthetic mRNA. And I'll kind of show why we're taking this approach initially. And then at 6 to 8 hours after, take the cells for gene expression by RNA-seq and chromatin accessibility by ATAC-seq at 48 hours. And so the idea here is that when we introduce this EWS fly by mRNA, it's going to be a rapid introduction, and genes that change expression very early in the 6 to 8 hour time point has been shown for other transcription factors. We'll be able to capture the genes that are directly targeted by EWS fly. Of course, we'll be confirming that down the road, but it's going to give us, it's going to facilitate the ability to do this in a large amount of cell types. And then, of course, the later time point, we'll start to see the chromatin changes, and that's where we're going to be using ATAC-seq. And as I mentioned, the reason why we're using the mRNA is that we can achieve very high efficiency of transfection in neural crest and mesodermal MSCs, here using a construct that co-expresses GFP. And we can also titrate this as well. So it gives us a high degree of control. It's very inert, non-toxic to the cells. And so this is the plan for our phase 1 of this approach. And I should mention that this is going to be done in individuals with minimal admixture, so mostly of 100%. And the idea here is to nominate some of the putative ALDR loci. And then in phase 2, following on from that, with our candidate ALDR loci identified, the plan is to stably activate EWS fly in subjects of unadmixed European ancestry, and then admixed African and Amarin ancestry, where the local European and non-European ancestry differs at these candidate ALDR loci to the best of our ability. And beyond that, looking at sort of like mixed ancestry in this stage. And so one of the things that we had to overcome here is, how do we stably activate EWS fly in these developmental intermediates? And a number of groups have used constitutive systems like virus to do this, but we wanted to sort of as closely replicate the real EWS fly translocation as possible. And so to do this, we developed another acronym, which is the conditionally activated somatic heterozygous EWS fly, or CASHUs. And so what this system is actually, it uses CRISPR-Cas9 to induce a translocation between EWS-R1 and FLY1, which we had demonstrated previously. But in this case, what we do is capture a gene expression cassette here, or a gene trap cassette is the appropriate terminology, that contains a splice acceptor and a strong stop site, and a polyadenylation, and then a selectable marker. And so what this does is it captures the translocation, but it doesn't allow it to be expressed. following administration of Cre recombinase, you get a recombination event between the two LOXP sites. The splice acceptor is removed, and now the fusion protein is able to express. And so we've, in proof of concept, shown that we can do this in iPS cells. So this is just looking at junction PCR at these loci, verifying that we have a left and right junction and its sequences at the translocation. And then in these iPS cell lines, we see that they express GFP as would be expected by our GFP cassette. And then when they're treated with Cre, we see a rapid loss of GFP. And so this is something we're still in the process of validating all of the points of this, but we think that this is going to be a really unique tool to be able to look at stable induction of EWS-Fly on these different ancestries, which is how we will go about the work in phase 2, where we have admix lines, we install cashews, and then generate these intermediates that are expressed in EWS-Fly and carry out a battery of functional assays as well as molecular assays to really confirm our final sort of nominated alder loci. And once we have those, the next steps will be to validate them using CRISPR. So for instance, if these alder loci are near genes, we'll look at overexpressing or knocking out the function of those genes. And then also looking at doing some long read sequencing to characterize the properties of the GGA repeats lengths around these loci. And so a lot of this work is still in progress and is currently being conducted. On the developmental side, so what I've shown is we've successfully established the methods to produce these neurocrest MSCs and mesodermal MSCs from iPS cells. We've carried out the replicate differentiations in multiple iPS cell lines, successfully carried out the ATAC-seq protocol, and the sequencing is underway. And so the next steps here really are the bioinformatic analyses and then the downstream validation. And then on the genetic ancestry side, we've identified and obtained a preliminary cohort of these ancestry lines, and we've diffed these to neurocrest and MSCs, and have established methods for the transient and stable expression of EWS-Fly. And so the next steps here, what we're doing is identifying and obtaining the additional ancestry lines, as well as beginning in the phase 1 experiments. And so, as I said, a lot of this is a work in progress, but I think it's going to really generate some exciting data down the road. And I'd certainly be interested in hearing anybody's thoughts about, characterization and just their general opinion about the approach and any ideas. So with that, I'll end and acknowledge all the people in the lab, especially Kelsey Becklin, who's a graduate student and leading most of this work, Rebecca Madden in my lab, who does a lot of the iPS cell cultures, some for Ewing, a lot for the other osteosarcoma projects, and the other members of the lab, Garrett and David's lab, Logan and his lab, Lauren Mills, who's helping with the bioinformatics, and our funding sources, which includes Ewing Sarcoma, which really got this whole thing started, especially on the ancestry side. So I'll end there and take any questions.
I'm just going to take MC privilege to note that Beau and I are so excited about especially the potential of looking at cell lines of different ancestry to identify the critical transcriptional targets of EWS-Fly1, that we pushed it to NIH, and it's getting reviewed by study section as we speak. Well, I don't know about the second, but it's getting reviewed now. So anyway, exciting work. But I know this work very well, so I shouldn't take up the questions.
Yeah, it looks like let's see if I can maybe stop sharing my screen.
Well, I'll ask this, Beau. Translocations are very common in sarcomas. Is your cashew system? We'll have to change the acronym, or at least make it a backronym where it means something else. But you could adapt it to other translocations. Is that right?
Yeah, absolutely. And I think, once we vet out the system, that's something we're definitely interested in doing.
Yeah, and in addition, have you tried differentiating iPSC into some of the other tissues of interest in sarcomas? Can you make fat, cartilage, etc.?
Yeah, so from the MSCs, using different differentiation protocols, you can generate, as I showed you, the osteoblast, which for osteosarcoma is very interesting to us, and then also definitely adipocytes and chondrocytes as well. There are protocols to generate those quite effectively.
Fantastic. I think we'll move on to the next presentation, assuming that the Drs. Skubitz are now available. I don't know which one of you is presenting. You're both listed on the.
We're going to try to do a tag team. I'll start and Amy will cut in.
Okay, wonderful. So I will introduce you then. So Dr. Keith Skubitz is an oncologist in the Department of Medicine, and Dr. Amy Skubitz is a researcher in lab medicine and pathology, and they are jointly going to present on predicting relapse and response to chemotherapy in sarcoma.
So hopefully you can see it and hear me.
Yes, I
Great.
can.
Let's see. Okay, how do I advance them? Down here. Okay, so this is just an outline of what we're going to talk about today. We're going to kind of review the importance of heterogeneity of sample sets, even though I'm sure you guys all know of that, but it's really important. Then we're going to talk about the use of gene expression to identify some of this heterogeneity, and maybe use that to predict recurrence risk in sarcomas. And then Amy's going to talk about the Ewing Sarcoma funded stem cell project, which went along with Dr. Cheng's PET study. And then we're going to come back and review giant cell tumors as a model of paracrine signaling and talk about how targeting macrophage recruitment may be very useful in sarcomas. So the key points of the whole talk are that heterogeneity within a diagnostic category is critical, that tumors are composed of both host and cancer cells, and that infiltrating inflammatory cells may be good or bad. As you know, sarcomas are heterogeneous. I think the WHO has about 150 subtypes of sarcomas now. The problem is, even with a histologic subtype, within that group, there's heterogeneity, and this complicates both clinical care, the interpretation of clinical trials, and drug development. Just to kind of possibly beat a dead horse, but if we have a clinical trial showing a drug A compared with a control group, and we look at a survival curve, some people might interpret this curve as being not very different, and others might say this is a major effect. But if there is heterogeneity there, for example, let's just assume that the treatment has no negative effect and it affects half of the patients. That means the actual benefit is twice as great as for the pooled group, and for the other half of the population, they achieve no benefit. Sometimes this is evident in clinical care because if the tumor is getting smaller, the drug would presumably be working, and it could be quite helpful for some time. In contrast, if the drug isn't working, obviously it isn't working. So it would be good to try to predict some of this heterogeneity early on, and we questioned some years ago whether we might use gene expression to do that. Now, one way to do that is to take a group of patients and say these guys have bad disease and these guys have good disease, and try to, look at differences in gene expression between those groups. It sounds straightforward, but it's not so obvious. The problem is, how do you define the two groups? And if you don't do it right, you're also going to have irrelevant genes, obscuring patterns, and different genes could be important in different subsets. For example, let's look at this survival curve of a group of patients. I think we would probably say these guys who are alive out here at a long time, they must be good. And someone who dies very early must be bad. But where exactly do you draw the line if you're going to form two groups for comparison? Do you do it here and here? But then you have very few patients. And you're also may not be incorporating these patients, or do you do it here and here? So The idea of forming two groups, good and bad, is not so straightforward. If we had a limitless number of patients, it would be easier. But in sarcomas, we have a finite number of patients, so we have to be careful how we do that. So we questioned whether we might be able to identify subsets independent of clinical information. If we just take a renal cell cancer as an example, which is true of any cancer, if we have a normal cell over here and you get a few mutations, at some point you have kidney cancer. Now, this is kind of a gradual progression, and in some cancers you have lots and lots of, for example, in smokers, maybe 30,000 mutations. There are other tumors where whole genome sequencing shows only maybe 100 mutations, only one or two of which may even potentially be relevant. So there's going to be quite a mix as cells progress to become more malignant in terms of how many and what mutations exist. And there's, like, as I say, there are likely many different combinations of mutations, leading to a potentially a very large number of types of cancer if we looked on a genetic basis. So one approach would be to look for a mutation or a small number of markers that might predict biology, which is perhaps reasonable. But I think we have to view the effects of a mutation are going to be manifest in the setting of the total background biochemistry of the cell. We could, for example, view the cell as a bag of water with billions of chemical reactions. The products of one reaction might be the reactants of another reaction. And then you have subcellular compartmentalization. So I think it becomes extremely complex. So in our early studies, we took the simplistic approach of comparing kidney cancer with normal kidney, and we found a lot of genes that were different. But then we decided to compare it with diseased kidney, and we found, not surprising, that the diseased kidney is different from normal kidney. And many of the things that we found in kidney cancer were actually also found in diseased kidney and appear to be indicative of inflammation. And at first one might think that inflammation is a good thing, but perhaps it's actually a bad thing. And then the question is, which cells are expressing the genes when we're measuring it? Is it the tumor cell or the normal infiltrating cells? So how could we approach this? So we thought we would do the following approach. We took 3 different types of tumors: kidney cancer, ovarian cancer, and the sarcoma aggressive fibromatosis. And we said, well, let's see if there are generally 2 big groups or subsets of these diseases that we could identify. And the way we did it was we formed a number of gene sets that represent known biochemical pathways, like OXPHOS, glycolysis, protein synthesis, DNA synthesis, things like that. And then we said, if we can really identify 2 general groups of these tumors, perhaps these different pathway genes would identify them, and presumably the different pathway gene sets might identify the same groups. So it turned out that indeed, we could find 2 global patterns of expression in kidney cancer, ovarian cancer, and fibromatosis using this technique. That led us to believe that we were perhaps actually finding 2 different subgroups of these diseases. So we said, what genes are most differentially expressed between the 2 different groups of kidney cancer, ovarian cancer, and fibromatosis? And we call these gene sets by the appropriate name. And then we questioned, well, if this is true of these tumors, could we use these gene sets to identify major subgroups of sarcomas? And in some of our early studies, we thought that yes, we could. And we still didn't know if this was due to the intrinsic differences in the tumor cells or in the host response or both. But we didn't actually have enough clinical information to see if this was a useful concept. But we were able to get some data from other people, and we asked the question, could these gene sets identify subsets of specifically high-grade pleomorphic soft tissue sarcomas that would have different biology? The first data we got was from Lund with 73 high-grade UPS, basically, tumors. Unfortunately, they had made their own chips, so we didn't really get all the DNA information that we had in our gene sets. So what we had to do was pool our gene sets and create one pooled gene set. And we did hierarchical clustering of those 73 samples. Now we got clustering, obviously, because that's what happens when you do clustering. So we questioned whether this first branch point sample set was different from this sample set in terms of probability of developing metastases. These patients did not get adjuvant chemotherapy. And indeed, they were different. So we were kind of pretty intrigued by that. But remember, I said we don't think there's just two kinds of sarcomas out there. There's potentially an infinite number. So if we could, from a practical point of view, get a few more than two groups, we might get better differentiation. So we went back to the clustering and said, what about taking the next branch points, therefore forming 4 groups? We had one problem here. We don't have many samples in this group, not enough for statistics. So we just looked at 3 groups, and we forgot about those guys. And again, we got perhaps a better separation of the groups. Didn't have all the data from that data set, and we only had 73 samples, but it was hopeful. So we next got another cohort of 300 samples from France, and we had all the data sets there, so that was great. We did the clustering again, and if we looked at 2 groups, there was a clear difference. This may not look as dramatic, but this is actually a 15-year follow-up, so it's pretty impressive. And then we looked at the 4 groups, and we found a very nice differentiation. So here, where almost everyone recurs, this clearly is, from a practical point of view, a very different group from one in which 20% of the people recur. And that would be important to think about in terms of both designing trials and actual clinical care, and questioning what is the biologic difference among those groups. That was a mixture of pooled soft tissue sarcomas that were high grade. In this case, we had enough samples that we could, we could actually take dedifferentiated liposarcomas and try to look at those. And I don't know if you're getting a distraction on the top of your screen, but it's really quite striking. Only about 5% of these people recurred, and more like 65% of these people recurred. With the UPS samples, again, a significant difference, and we could fragment the UPS guys into 4 groups as well. So, and the p-value was obviously sizable. So I think both of those studies confirm the idea that this approach is quite reasonable for regular soft tissue sarcomas. We were also able to get 60 samples again from the French group of GIST tumors, which are a bit more homogeneous. And using, for example, the ovarian cancer set, there was a marked difference in recurrence risk between our good and bad prognostic groups. Now, with this particular tumor, there are other ways that you can predict recurrence pretty well. We call it the Meitner risk score, and there's something called low, high, and intermediate. This is important because these people now get adjuvant chemotherapy, which is fairly expensive, potentially $100,000 plus per year, and they probably would wind up taking it at least for 5 years, if not permanently. So if we could find some of these people who didn't need to get treated, that would be a good thing. So we took this group of patients and pooled these, and then did our hierarchical clustering with either the fibromatosis gene set, the ovarian, or the kidney, or a pooled set. And we were able to get a group of patients who don't recur, suggesting that we could have these people avoid a complicated and expensive treatment. So I think that at this point, we think that some biological behavior that doesn't correlate with morphology, we could perhaps determine by gene expression patterns, and we're hopeful that this will have further application. So at this point, I was going to switch to Amy, and she will talk about another study.
Hi, well, this is a study that was funded by the Ewing Sarcoma Foundation, and it was basically looking at the density of cancer stem cells within a patient's soft tissue sarcoma. And our question was whether or not this density could actually predict how the patient would respond to treatment. And just as a background to define cancer stem cells, they're a small population, typically 1 to 10% of the cancer cells within a tumor. And they have stem cell properties, namely the ability to keep dividing and self-renewal, but also they can perpetuate tumor growth by giving rise to the bulk of the tumor cells. And the important thing is that they may resist standard chemotherapy, so they're really serving as a reservoir of cancer cells that may cause relapse after surgery, radiation, chemotherapy, such that the physician really thinks they eliminated all observable signs of the cancer, but these cancer stem cells still remain. And the thought is that if you wanted to cure cancer, you really needed to eliminate all of the cancer stem cells. So our question was, does a change in cancer stem cell density with treatment correlate with outcome? And for this study, we really needed a clinical trial. And we were very fortunate that Dr. Chang and his colleagues had a clinical trial ongoing at the time. And their study was looking at the PET CT scan response after neoadjuvant chemotherapy, and to see whether that predicted progression-free survival in a prospective trial for soft tissue sarcomas. And the way the trial was arranged was basically the patients would have a PET CT scan, and at the same time a biopsy, a very small biopsy, would be taken of the tumor. The patients would then undergo chemotherapy, typically for about 4 months, and then the patient would again have a PET CT scan, and then surgery of total debulking of the tumor would occur. So we used this basically to test our hypothesis. The hypothesis of the study was basically that the number of cancer stem cells in the residual tumor of the patient, so in that big piece of tissue that was removed at surgery, actually would predict the treatment outcome. So if there were very few or no cancer stem cells present in the tumor after treatment, then the patient would have a good outcome. So that was our hypothesis. And fortunately, as I mentioned, there was biopsy samples as well as surgical samples from the patients. And although I believe over 60 or 70 patients were enrolled in this clinical trial, there were 31 sarcoma patients who actually had a sizable amount of biopsy and surgical sample that we could then conduct this study doing immunohistochemical staining of the tumors from these 31 patients. And the markers that we decided to use for the immunohistochemical staining for the cancer stem cells included ALDH1, which is aldehyde dehydrogenase, CD44, and CD133. And these are 3 markers that others have used looking at cancer stem cells in a variety of other types of cancer tumors. So we felt these gave us the best chance of working in our study. We also had the angiogenesis marker CD31, which marks endothelial cells or blood vessels. And then you'll see later we also included the macrophage marker CD68. And the pathologist who worked with us on this was Dr. John Wilson, and he helped develop a scoring system for each of the markers. And it was really based either on the intensity of staining, so you get a score of 0, 1, 2, or 3, or the percent of tumor cells that stain positively. So here's an example of how he came up with his scoring in the 0 to 5% up to over 75%. And so I just wanted to give you one example of how the sarcomas were scored for angiogenesis. Here's an example of 3 different tumors, and you can see the blood vessel density is scored either 1 on the first one, 2 on the second, and then a score of 3 on the third, so that there's lots of blood vessels staining there. And then when we went ahead and stained the tissues for ALDH1, looking for cancer stem cells, Dr. Wilson pointed out that the staining that we're seeing in this tissue, this is just one of the examples, was actually not the tumor cells that were staining, but it was the macrophages. And this was quite a disappointment, as you can imagine. So he gave this one a score of 0 because no tumor cells actually stained positive for ALDH, or they stained very, very faintly in some of the cases. So this is just a summary of years of work, and basically just to show you the data is that ALDH1 stained positively in about 11 of the 30 cases, but there really was no change pre-treatment and post-treatment. CD44, 26 of the 30 stained positively pre and post-treatment, and then CD133, the very well-known cancer stem cell marker, was only positive in 2 of the cases, and that was pre-treatment. So the fly in the ointment here was that ALDH1 and CD44 actually were the macrophages were what was staining positively in the sarcoma tumors. So this is just to show you, we then went back and stained the tissues again with this macrophage marker, CD68, and you could see here like over 25 to 50% of the cells were positive, and again, unfortunately, those were the macrophages, which were really heavily infiltrating the sarcoma samples. So this led us back, to our hypothesis, and basically we weren't able to actually answer our hypothesis because we didn't really have a good cancer stem cell marker for sarcomas. We would just caution everyone not to use CD133, ALDH1, or CD44, since they did not appear to be good markers for sarcomas, and really points out the concept that if you have a marker for cancer stem cell, it might depend on which tumor you're staining. We were, however, very excited about the fact that there was prominent macrophage component in sarcomas. It was very striking, and what we found is that the amount of staining in the pretreatment sample, it ranged from less than 5% to over 50%. And the point should be that macrophages have been recognized even as early as the 19th century to infiltrate tumors. So our study, the baseline macrophage density really correlated with the baseline PET CT scan intensity, and also it correlated with the percent tumor kill post-chemotherapy. So we really feel that macrophage infiltration may influence the response to chemotherapy and provide a potential target of therapy. And just to now segue into Keith, I'll let him talk the rest, but chronic inflammation has been linked to cancer development. We know that macrophages secrete cytokines that are capable of paracrine signaling, and that they could play an important role in sarcoma biology. So I'll let Keith take the next part.
Okay, I just wanted to talk about paracrine signaling in sarcoma and give two really interesting examples in case you're not aware of them. The main question is, what are the host cell tumor cell interactions here? As you know, in autocrine signaling, a cell is releasing a factor that feeds back upon itself, whereas in paracrine signaling, the cell is releasing something that feeds back on a neighboring cell. Now, giant cell tumors are a type of sarcoma that have been reported to produce factors that attract osteoclasts and their precursors. And when metastatic, it tends not to respond to traditional chemotherapy. This is what one of those tumors looks like. These big cells are osteoclasts, and these arrows point to the actual tumor cells. Sometimes you might not have that many tumor cells in here, but there's a significant number in there. However, they look quite similar to some of the precursors of these cells. So there's cells in here which have been recruited into the tumor by the actual tumor cell, and these cells will eventually fuse to form these big giant cells. So the model is the giant cell tumor produces something. We happen to know what it is. We and others described that with Drs. Chang, Ogilvie, and Thompson a little while ago. And this thing attracts osteoclast precursors to form osteoclasts. So the question is, could we use an antibody to that rank ligand as a treatment of giant cell tumors? The thinking there was based on how bone remodeling works. We know osteoblasts make rank ligand, which stimulates osteoclasts. We know when bone is remodeled, there's a wave of osteoclasts moving through dissolving bone, followed shortly thereafter by osteoblasts, suggesting that osteoclasts are making things that are trophic for the osteoblast. If that model were true for giant cell tumors, if the osteoclast is making something that the actual tumor cells like, and we block this, we should block this, and we should block this, and this cell may or may not like it. So this this reasoning led to a phase 2 trial of an antibody to rank ligand in giant cell tumors, and it worked quite well. This is one of our early patients showing a lung metastasis 5 weeks after treatment and 37 weeks after treatment. In fact, this person, she actually drove here from Ohio monthly, showing total dissolution apparently of part of the vertebral column with it's impinging in the spinal cord. She had quite a bit of pain in her left arm and hand and couldn't move it well. After 4 months, the bone actually resorbed. And she's quite adamant that the pain actually started improving in the drive back to Cleveland, which is, a bit more than about 12 hours. Those early studies led to a trial of 500 patients, and it's clear that almost everyone got clinical benefit. The really interesting point is shown down here. The tumor cells themselves actually disappeared, even histologically, even though the drug is not affecting the tumor cell. So that's really, I think, quite fascinating. By getting rid of a normal infiltrating cell, which is recruited by the tumor cell goes away. We think this is a classic model, and I think it's the first one described in humans for targeting a paracrine autocrine signaling pathway in cancer. There's another tumor that we see a lot, another type of sarcoma. It's got a long name, abbreviated PVNS. In this case, the tumor cells, sometimes there can be very, very few as 5% of the tumor are actually tumor cells. And they actually produce something called CSF1, which recruits macrophage precursors, and the tumor is infiltrated by macrophages. Dr. Cheng was actually involved in a study of an antibody to CSF1, which worked quite well. And again, the interesting point is that in some patients, by targeting something which affects recruitment of normal cells, not targeting the tumor cell itself, the tumor cells actually go away. I guess I got ahead of myself there. Dr. Cheng did that study, but here's the first description from France showing this tumor here. The tumor goes away with an oral CSF1 receptor inhibitor. We actually hope to begin a new trial here soon with a new drug which inhibits CSF1, which for various reasons we won't go into is a very attractive one. So the real question is, would this be useful in sarcomas? Getting back to the heterogeneity concept, we don't want to just treat all sarcomas with this. We want to actually know there were a significant number of macrophages present in the tumor that we could affect. And it will be very interesting to see what that shows. Now, of course, macrophages are felt to be important in the immune response, and that could be very impressive. But there's no reason to think that, analogous to our other two sarcomas I just described, that in some cases, possibly getting rid of the macrophage might actually affect the tumor without having to invoke the immune system. I think we just need to see where that goes. Well, we would like to thank Ewing Sarcoma and multiple other funding systems, along with a whole bunch of different collaborators, specifically Drs. Pambuchian, Manivel, Cheng, Close, Ogilvie, and Thompson, and Ben Meisner, who was a medical student, and basically all these other guys as well. So I think we've actually kept it under the allotted time, and we're available for some questions.
Thank you both. I think we already have shown we have a shy crowd with respect to questions, but I'll give people a minute to speak up.
So Logan, I have a question, if I may.
I don't think the question's for me. It's for one of the Dr. Skubitzes.
For the Skubitzes. So are you concerned that clustering based on small gene sets may not be replicable across data sets? Built on different platforms or different tissues?
Could be, but I mean, our gene sets are actually about 300 or so genes, depending on how we do it. I think there's, just based on the concept of a global gene expression pattern, there's going to be lots and lots of different gene sets that probably would work, and wouldn't have to be this one. But I think we have 3 different data sets that seem to reproduce that phenomenon.
I guess as a follow-up question, do you think there's something specific about those genes, or you could choose sort of any 300 genes, and these might represent patterns that are present in a small percent, in the data set, and then you'd be able to recreate similar data?
I think that there's not necessarily anything magical about these genes. In fact, each of the 3 gene sets we have, which they're almost mutually exclusive, there's like 5 genes that are shared, they all will separate pretty much the same 2 groups, maybe not exactly. That's one of the things that has limited this technology, is the concept of intellectual property and what company wants to develop it, because if we have this gene set patented, there's going to be some other ones that will probably be just as good. I think right now the main mover there is the French group, because they have a very organized system. Virtually everyone in France with a sarcoma is part of their trial, and they're trying to reproduce this system with something called SynSarc, which is around 70 genes. Interestingly, most of which are not present in our gene sets. I think well, yeah. So I think that there's nothing magical about these gene sets, because there's just so much biochemistry happening in the cell. You can take snapshots of different parts of it, and you may be able to get similar groupings. If you get down on a finer scale, that probably isn't true. But if you just wanted, for example, to see which cells are more metabolically active, you might get a grouping there. The whole point here, though, I think, is we can get better than just good and bad, which is not, I think, acceptable. I think we need to really identify a lot more fine specificity with multiple different groups that are going to have different properties. I don't know if I answered that question, but
no, I think you answered it pretty well. I would love to. I'm going to discuss later in the talk today some approaches that are relatively automated and unsupervised for doing very similar approaches on full gene sets, and would love to run your data through and see if it comes up with similar approaches.
Sounds good.
Thanks.
So I had a question about methods for Amy. So in the work that you described at first, Amy, you were saying, well, we didn't find good markers for cancer stem cells. And so I was wondering what you think about a functional assay for cancer stem cells that could have been used. And do you think we could study cancer stem cells fruitfully using patient-derived xenografts? So for various sarcomas, we have those here at the university. And I wonder if we try to functionally define cancer stem cell using the patient-derived xenografts, how valid you think that will be, since it'll be a cancer stem cell in an immunodeficient mouse, but that may not mean the same thing as in human. So it's sort of a practical and philosophical question.
Okay, well, I'll just start out. The 3 markers that we chose were ones that there were antibodies commercially available, and the tissues were all paraffin embedded. So we were kind of limited in that respect to what we could be examining. And there was just a very limited amount of tissue in the biopsies. And again, they're formalin fixed. So we really couldn't be doing anything functional. So what you're proposing, using actual live cells, sounds much more exciting. And maybe those markers that I said didn't work that well for us may work in your system. But I still think if you use the same antibodies, I think you're going to have the same problem. Like if you were sorting out the tumor and tried to sort it using these antibodies, you might pull out a lot of macrophages in the process.
Right. So I also had one question for Keith, if that's okay, which is, I thought in PVNS that the tumor cells were also expressing CSF1 receptor. So if you use a CSF1 receptor inhibitor, I suppose it may be targeting the tumor resident macrophage or infiltrating macrophage as well as the tumor cells themselves.
Yeah, I'm not actually sure of if they are expressing that receptor.
Okay.
Maybe you actually know that. I know the early studies had a lot of difficulty finding or reproducing the translocation, because you could have as few as 3 or 4% of the tumor, or the cells in the tumor were the actual tumor. But when they looked at RNA and protein expression, I mean, they were almost all of them were making the CSF1.
Okay. Yeah, I understand the translocation is present in the minority of the cells.
Well, it's present in the tumor cells, but not the macrophages.
Okay. Yeah, I guess I'm looking at a paper from Matt Vanderman, but it's very confusing, I guess, about what cells are really making.
Is that a PNAS one?
Yeah.
Yeah, yeah, I think does it actually say they felt the PDNS cells themselves had the receptor?
Well, it's not clear. Like,
oh, yeah, I think the concept was that the history of that was that some people reported a translocation and other people said they couldn't find it.
Okay.
And then when people looked really closely, they still had difficulty finding the translocation. When we looked at mRNA and protein, they could find it in almost all of the cells. But the bottom line was that when they could find it, sometimes the number of tumor cells were as few as I say, 3 or 4% of the actual mass of cells, which is why people couldn't find the translocation. And all the other cells were macrophages or macrophage precursors.
Okay. We know some osteosarcomas make it the receptor. And so we were interested in it as a target in osteosarcoma. But then they also recruited a lot of macrophage. So it'd be interesting to think about the same approach for osteosarcoma.
Right. I think the one issue here is like, again, getting a very limited sample set of specific disease that you can interest a company in supporting such a trial.
Our speaker is Brittany Siontis, who's a medical oncologist at the Mayo Clinic. She earned her medical degree at the University of Washington in Seattle. She's active in research and education with a focus on evaluating novel therapies for bone and soft tissue sarcomas through clinical trials, and she's going to tell us today about her work on using tumor cell-free circulating DNA as a biomarker in sarcoma. So thank you, Dr. Siontis.
Thank you so much. Can everyone hear me okay?
Yep.
Great. So despite all of the Zoom meetings that we've had in the COVID era, I'm still not the most tech savvy. So bear with me while I work to share my screen here.
Looks like it's working fine.
Great. So today, I would just like to thank Rain Sarcoma and the University of Minnesota for hosting this, and I'm looking forward to sharing with you guys my work on circulating tumor DNA as a non-invasive biomarker in sarcomas. There was some research support provided by Rain Sarcoma Foundation for the work that I'll be presenting today, and just an additional disclosure that's not particularly relevant, but I do serve on an advisory board. So kind of my goals for today, we'll just touch briefly on epidemiology. Obviously for all the clinician scientists, this is well known to all of us, but I was anticipating and hopeful that we would have nice attendance from our patient and caregiver populations. So just to share a little bit more background into why I'm doing what I'm doing. I'll touch a little bit on conventional imaging in sarcoma, current recommendations for disease monitoring, as well as the challenges we face in using conventional imaging in sarcoma. And then really touch on circulating tumor DNA. What is it? How is it being used in other tumor types? And how are we using it in sarcoma? So just to look at the numbers, in 2020, the American Cancer Society predicts more than 1.8 million new cancer diagnoses and over 606,000 cancer-specific deaths. And we all know that sarcoma makes up a very small proportion of that population, but it is still incredibly important to all of us who are on this call today. We know one of the great challenges in sarcoma is the heterogeneity within the disease, not only within the location of where these tumors occur, but the fact that there are an ever-growing number of subtypes of sarcoma, which makes it challenging at the time of diagnosis and can make it very challenging with respect to therapy. We know that to make an accurate diagnosis of sarcoma, it does require expert pathology review, and studies have shown that about 40% of diagnoses may be changed either from a non-sarcoma to a sarcoma or from one sarcoma subtype to a different sarcoma subtype once reviewed by a bone and soft tissue pathologist with specialty in these diseases. Just looking at how we approach surveillance for these tumors, we have our kind of national comprehensive cancer guidelines here in the United States, and then I did include the European Society of Medical Oncology guidelines as well, and for the most part they mirror each other, but really we're looking at pretty routine clinic exam, with physical examination and imaging very regularly for those first few years, and slowly spreading out over time the further out from definitive therapy we get. Here in the US, we use a lot of CT scans of the chest to assess for pulmonary metastases. In Europe and in Canada, they do a lot of chest x-ray. We're looking at MRI if the primary tumor was in, the arm or the leg, and we're doing CT scans of the abdomen and pelvis. We know that there is a lot of limited sensitivity to our physical exam, so we rely very heavily on these images, and it does lead to not only radiation exposure with CT scans, but significant financial toxicity for our patient population. We also know that imaging is not always perfect, and it doesn't always give us the best assessment of whether our patients are truly responding to chemotherapy or not. And we are limited in response assessment to something called RIS. And again, I know for all the clinician scientists, this is well known to you guys, but for the patients and caregivers on the line here, really this is a matter of, us and our radiologists measuring the tumor on the CT scan or on the MRI and looking at the change in that number over time. And if the accumulation or the sum of all those tumors that are being measured over time increases by greater than, that's a typo, it should be, is less than 30% or a greater than 30% decrease, then we say that it's a partial response. If that sum increases by more than 20%, then we know that that's progression of disease. But this can be really challenging in sarcomas, which don't always shrink reliably on scans in response to treatment. So this is an example of a patient who had metastatic gastrointestinal stromal tumor. And we can see their baseline scan here with multiple tumors within the belly. They started imatinib therapy, and based on that RECIST criteria, there was a significant increase in size of those tumors. But when we actually went back and got a PET scan, we can see that while the rim of that tumor is still lighting up, the middle of that tumor is what we call cold, or it's not very active. And that's a sign of necrosis. So if we had just based our decision on this scan, then we would have said, gosh, this patient is progressing. We need to move on to the next line of treatment. But with evidence of necrosis or cell death, then maybe we have more of a signal that this patient is actually responding to this imatinib therapy. Another example is a patient with a large undifferentiated pleomorphic sarcoma in their leg. And it was a little over 11 centimeters at baseline. And we did a repeat MRI after 2 rounds of chemotherapy with doxorubicin and ifosfamide. And that tumor got bigger. But I think we can all appreciate that it looks pretty different here compared to the pre-treatment scan. And there was a lot of bleeding or hemorrhage within that tumor. And the question then becomes, well, is it growing and bleeding within the tumor because it's just growing and it's progressing and the chemotherapy is not helping? Or is some of that growth related to bleeding or hemorrhage within the tumor because the chemotherapy is actually leading to cell death? So this, constant dilemma in clinic is really what sparked my interest in looking at other ways to try and determine whether or not patients are benefiting from chemotherapy. And I started looking at circulating tumor DNA. And basically, circulating tumor DNA is DNA from the cancer cells that have been released into the blood of patients. We know that normal, healthy tissues undergo a process of cell death called apoptosis, and that normal cell DNA can be released into our bloodstream. But the same thing can happen with cancer cells. They can undergo apoptosis or necrosis either as a normal part of their growth process or in response to chemotherapy. And that tumor DNA is then released into the blood, which we can then capture with a blood draw and test for. This is something that has been investigated in many other different types of tumor. And I know this is a busy slide, but we can just see here that there have been many studies done looking at circulating tumor DNA in colon cancer, breast cancer, ovarian cancer, just to name a few. But I think one of the most important things to think about when thinking about this table is Most of these cancers have a tumor-specific alteration that is present in the majority of those tumors or those cancers that can reliably be tested for. And we can't really say that in sarcoma, right? Going back to that issue of heterogeneity. So how can we sort of use this clinically? This is an example of lung cancer. And we know that certain types of lung cancer have a mutation in something called EGFR. And this first graph shows in the black line a decrease in tumor size in response to a drug called gefitinib over time. And we know that the EGFR mutation that they were testing for in the blood also decreased over time in response to treatment. So this is a situation where circulating tumor DNA decreasing in the blood correlated with that patient having response to therapy. This second graph can also be very helpful. Again, we're looking at a specific mutation in this blue triangle here, and when treatment is started, that quickly drops off and remains undetectable. And initially, that correlates with a decrease in size of the tumor. But then, gosh, we see this tumor is getting bigger again. What's going on? We know that cancer cells can develop resistance mutations. So these investigators looked at a different EGFR mutation in the blood, and that was rising, signifying an increase in tumor DNA as that tumor was growing again. Again, this is very challenging in sarcoma. We don't have one disease with one genetic alteration that we can consistently test for in every single sarcoma patient that walks through the door. So it is very important that we take a more personalized approach to how we're going to be detecting and monitoring for this circulating tumor DNA. This has been investigated in sarcomas to date, and this is an example of several patients with gastrointestinal stromal tumor who were receiving various doses of imatinib therapy. And we can see in those patients a drop in detectable circulating tumor DNA in response to treatment. And in fact, in one patient, we saw a complete drop such that it was undetectable, and scans actually showed a complete response to therapy. But yet again, GIST happens to be a sarcoma in which we can reliably detect an alteration in KIT. So many of these patients are going to have a specific alteration we can look for. Similarly, in Ewing sarcoma, when we know that these tumors harbor a rearrangement in EWSR1, this was a study done out at the University of Utah, looking at about 96 patients with Ewing sarcoma. About 65% of those patients had localized disease at the time of their diagnosis, and those are the patients that are represented in these graphs. And Dr. Schulman showed that patients who had detectable circulating tumor DNA at the end of their treatment did worse. So those patients who had detectable circulating tumor DNA were more likely to have a relapse of their disease, and their overall survival was worse. And perhaps that reflects an overall burden of their microscopic disease. So how are we applying this to our patients here at Mayo? So we currently have a protocol looking at circulating tumor DNA in patients who have unresectable or metastatic sarcomas. These are patients who are planned to initiate chemotherapy, and for now we're just looking at the adult patient population. With our protocol, we do require tumor tissue, and we know that fresh tissue is always best. So we are utilizing tissue that's being obtained for a clinically indicated biopsy or surgery. Because we know it's not always feasible to get a biopsy, and we certainly don't want to ask patients to go through a procedure only for research purposes, we are in the process of exploring how we can utilize archived tissue for our testing. And essentially what we do with this tumor tissue is we do a type of genetic sequencing called MAPAIR sequencing, or MP-seq. And this allows us to do comprehensive whole genome sequencing of the tumor to identify changes in that particular patient's tumor, which we can then search for in the blood. With respect to blood collection, we are collecting 4 blood samples per patient who's enrolled. And really the time points of that collection varies based on the type of sarcoma and the type of chemotherapy regimen they are receiving. But typically we're looking at a baseline or a pre-chemotherapy blood draw at the beginning of cycles 2 and 3, and then at the time of disease progression. And we are using this blood to do polymerase chain reaction testing, PCR, to detect that circulating tumor DNA in the blood. So as I mentioned, mate pair sequencing is a type of comprehensive genetic sequencing. It is slightly different than traditional whole genome sequencing, and it's a bit of a less expensive option. It still allows us to detect chromosome rearrangements and copy number variants, but the downside is that we are not detecting single nucleotide variants. And basically, DNA from the tumor is extracted and prepared as it usually would be for next generation sequencing. We use this mate pair library protocol, which I'll show in a moment, and then a lot of bioinformatics tools to finally get that genomic data. So mate pair sequencing basically uses something called circularization. So it takes a fragment of that DNA, 2 to 5 kilobases long, and basically forms it into a circle. So primers are put at each end and they're joined together, and then that circle is split. And we're capturing and sequencing what's on those primer segments. So when we look at standard whole genome sequencing, we're doing sequencing of these very small segments, which can be very time intensive and very expensive in order to cover the whole genome. But with mate pair sequencing, with larger fragments, we're still getting good coverage of the genome, but at less depth. So it does make for a more cost-effective way to get this information. And this is what we come out the other end with. This is a genome plot from mate pair sequencing, and this is actually from a sarcoma patient. And what we're seeing here is each of the chromosomes is listed here, including the X and Y chromosome. The blue dots are areas that we see indicate gains. The red indicate losses. And these magenta lines are really what we're most interested in. These magenta lines show us gene rearrangements. And it can be a gene rearrangement between 2 chromosomes. For example, this is a rearrangement between chromosome 11 and chromosome 2. Or it can be a gene rearrangement within a chromosome. So here, chromosome 2 has an inversion or a rearrangement within the chromosome. And it's these junctions or these alterations that we are looking for to decide which genetic alteration, which is present in this particular patient's tumor, should we be searching for in the blood. And we use something called a junction plot to help determine which of these magenta lines is maybe going to be the highest yield for us. So on this junction plot, it shows us the 2 breakpoints that lead to that junction or that chromosome rearrangement. And it is displayed here on the top and bottom. This happens to be an inversion within chromosome 7, so that's why the same chromosome is listed. And the number of lines here is the number of fragments of DNA which support this junction being present. So the more lines we see, the more often we're seeing this junction. And that means we think this is more reliable because it's something that we're detecting at high frequency. We also look at how these junction lines are scattered about the plot. And we really want the distribution from the top and the bottom to be similar. And we want it to be about the size of the fragment length. If we saw these lines scattered all over this map, then we wouldn't find that to be a particularly reliable junction to try and test for in the blood. And then once we do the PCR to actually look for the circulating tumor DNA in the blood, we get what's called a melting curve. And we run a positive control, which is this gray line here. And this is doing PCR on DNA from the tumor, looking for that specific junction. So we know this should be positive. Then we run the blood sample, and then we do a negative control with water, which should be undetectable. So to date, we have enrolled about 20 patients. Some of these patients were enrolled pre our current protocol. So there's kind of a span of anywhere from 1 to 6 blood draws. And we can see that there's a pretty big distribution in terms of the types of sarcoma that we have been looking at. Not surprisingly, leiomyosarcoma, liposarcoma, undifferentiated pleomorphic sarcoma are amongst the most common. About 55% of these patients had tumor samples obtained from biopsy rather than surgery. We have completed analysis on 8 patients, and we've been able to detect circulating tumor DNA thus far in 3. And we're working with these other 5 samples on a method to try and pre-amplify that DNA to improve our level of detection. So I'd like to just walk through a few patient examples, and I'm sorry, I know I'm supposed to stop talking at 20 minutes, but I'll apologize and ask for a little more time. But this is a patient who had a history of uterine leiomyosarcoma, had surgery to remove a 10-centimeter mass in the uterus. 1 month later, had a 4-centimeter pelvic mass on PET scan, which we can see here. Had 5 months of chemotherapy and then radiation to that pelvic mass, and then was on surveillance. Unfortunately, a few months later, developed multiple metastases in the lungs. This patient was enrolled in the protocol, and this is the result of their mate pair sequencing. And again, we can see a lot of these magenta lines, and the thicker this magenta line is, the more frequent we're seeing that alteration. So a lot of potential targets for us to look for in the blood for this patient. The baseline sample showed no detectable tumor DNA. Again, this gray line is the positive. This is the junction present in the tumor, and this is what was in the blood and the negative control. So nothing was detectable. The second blood sample was also undetectable. Again, this line that we're getting from the blood is not lining up with what we know to be a positive result. So this is undetected. But then on that third blood test, we do see that there's tumor DNA detected. We can see the peak of this line lines up with that known positive. Obviously at a smaller quantity, this is being measured in the blood as opposed to the tumor itself. And then again, finally at the fourth blood draw, that was still detectable and at an even higher level. If we look at what was happening with this patient clinically, as I mentioned, had a couple months of chemotherapy, had response to treatment, was on surveillance. This was the first blood draw, which was undetectable. And about that time, the patient had actually undergone surgery to remove a tumor. That second blood draw, again, undetectable, and we had no imaging correlates with these. However, that third blood draw that then became positive, we did have CT scan evidence of disease progression. And that fourth blood draw, a few months later, despite being on chemotherapy, continued to show progression of disease. So I think this was a nice example of we can detect this in the blood in a personalized approach. And in this situation, it did correlate with progression of disease. This is another example of a patient who originally had a large sarcoma in their thigh. They had radiation with chemotherapy, followed by surgery, followed by more chemotherapy. 2 years after that, developed metastases in the lungs and had surgery. And 1 year after that, as we can see by this representative PET scan, developed multifocal disease and was recommended to undergo chemotherapy. Unfortunately, I don't have their mate pair sequencing plot, but they did have a number of junctions for evaluation, and this is just one that we chose. This is on chromosome 10 and was an inversion within that chromosome. And there were 27 detectable fragments, which were clustered nicely, that displayed that inversion. And this is displayed a little bit differently, but we can see baseline in red. We can see their second blood draw in blue, and their third blood draw, the colors don't match perfectly, but it's in green. And the second blood draw correlated with initiation of new chemotherapy in response to treatment. And then when we saw that tumor DNA level go up again, that was again a sign of progression of disease, and that's just shown in a slightly different graphical way here. So where are we taking this next? Obviously, we still have a number of samples to be analyzed, and we continue to refine both how we're handling the tissue and how we're handling the blood to try and improve our levels of detection. We're interested in exploring other circulating tumor biomarkers, including fusion proteins or circulating tumor RNA. There has been some data looking at circulating tumor DNA in urine in other tumor types, and the advantage of doing urine detection is a much greater volume of sample can be obtained, and it is non-invasive for patients. And then also, how can we utilize this technology not just for disease monitoring to chemotherapy, but for disease surveillance? So those are kind of my questions and things I'd like to ask in the future. So I apologize again for taking a bit of extra time, but thank you for allowing me to do so, and I'd be happy to take any questions.
All right, thanks so much. That's fascinating work. That's open for questions. Maybe I'll just start with one quick. So I'm curious, you know, it's fascinating that you can use this personalized approach and find breakpoints that you can use. I was wondering whether you think another way to do it would be to use recurrent tumor-specific methylation changes to see if they were really specific for cancer cells, maybe, you know, and you use methylation sensitive PCR reagents, maybe that could be used in any patient.
Yeah, I think it's a really interesting question, and we've definitely spoken about methylation and how could we potentially use that as kind of a marker of response assessment. And I'll be very honest, I don't know a lot about methylation, so I won't embarrass myself by trying to speak about it at length, but it's certainly something that we've had discussions for. I think it's very challenging. I think obviously we want to try and have an approach that's going to be, you know, quick and cost effective and useful for a broad number of patients. But I think this disease just challenges that. I think we have to think a little more on a personalized approach.
Other questions?
So it looks like someone typed one in here. Have you had any patients whose cancer progressed and yet had no tumor DNA in the blood? So that's a really good question, and we haven't run into that quite yet. But you know, this is still in its infancy, and I think it's very possible. We're obviously relying on necrosis and apoptosis, you know, to detect this in the blood. We've certainly had patients that we haven't been able to detect it at baseline, but we know they have active cancer, so it's a challenge. We're still working on it.
Brittany, this is Brenda. Great talk. Thank you.
Yeah.
One of the challenges with this approach in the pediatric space is the volume of blood required, really limiting our ability to do this in the pediatric population, except for sort of more the older adolescent, young adult population. We're certainly trying, but do you have thoughts on sort of, and you mentioned the urine piece, which I would be very supportive. That's way easier.
Yes.
But thoughts about sort of smaller volume techniques, level of detection, the level that's meaningful, you know, ideas around that. I think the urine is interesting, but I think there's so much data still needed to actually think that that's going to be even reliable. Whereas blood, at least I think all of us believe we can detect something.
Yeah.
What are your thoughts around trying to scale this to small volume?
Yeah, it's really hard, and that's precisely why we have not enrolled pediatric patients, because right now with this protocol we're taking 40 cc's. You know, with the protocol we're thinking about in the future to look at DNA, fusion protein, and RNA simultaneously in patients, we're looking at 60 plus cc's, which, you know, we just can't do that in kids. You know, I think exploring these pre-amplification techniques could be really impactful in terms of being able to use a smaller volume of blood and still reach a level of detection that we think is reliable and believable. But we are still running into that technique even when we have 40 cc's of blood. So it's a problem.
It's a challenge, put it that way.
One we will eventually overcome, but.
Any other questions? All right, thanks so much.
Thank you very much for having me.
Sure thing. So our next speaker is Dr. Aaron Sarver, who is an assistant professor in the Institute of Health Informatics here at the University of Minnesota. He's been doing cancer genomics research here at the university for a long time. And he's going to tell us about his recent work in osteosarcoma.
I guess I better unmute myself before
Yeah, you should.
before I go to try to share the screen.
That looks good.
Okay, great. So I'd like to thank the organizers for giving me the opportunity to speak today, as well as the other speakers. There have been some great talks. So I've really been doing data science-based approaches to understand cancer genomics for longer than I really care to admit. My approach has always been to let the data generate the hypotheses, which we can then test and replicate in order to ensure validity. So essentially, I've been using what amount to AI-based approaches, and was doing this prior to the widespread use of the term artificial intelligence to describe statistical analysis. In order to do this, I've had to create a number of approaches that I'll try to describe. So we've been really using, as many other people have described today, a comparative genomic approach. So osteosarcoma is a rare and devastating pediatric cancer in humans. In canines, it is much more common than it is in humans, and especially in large breed dogs. So our approach has been to integrate the study of naturally occurring canine and human tumors with the study of lab-generated tumors in mice to try to understand the key molecular indicators of disease and disease progression. So the hallmark of osteosarcoma is genomic chaos. And as others have said today, for all the research that has been done in the field in recent years, patient survival has not improved during the genomic era. So into this talk, I'm going to give a history of some of the OS research that I'm involved with at the U of M and try to integrate that into a series of unpublished work that is ongoing. So my first involvement in osteosarcoma came in conversation with Jaime Mariano, who was really trying to understand the molecular basis of osteosarcoma in large dogs, where osteosarcoma occurs at very high rates. As many as 20% of Irish wolfhounds will contract osteosarcoma throughout their lifetime. Because primary OS tumors can be directly grown in culture, Jaime had a collection of low passage cell lines, which he had profiled using for the transcriptomics using the canine Affymetrix array platform. He also had outcome data for the dogs after treatment. And because these were low passage cell lines, the contribution of stromal tissue was removed from these transcriptional profiles, and this greatly simplified the complexity of the analysis. So we looked for associations between outcome and transcriptional patterns. And really luckily for us, the principal component of variation in the data was a set of genes associated with cycling cells, which included Ki67. So anyone in the audience with basic pathology knowledge sort of knows that Ki67 protein level in tumors is associated with rapidly dividing cells and poor outcomes. So when we look at the outcome in samples that had high levels of this cluster of genes, they were universally bad. And even with the limited sample power we had, they were clearly separable from the outcomes of animals that had low levels of cell cycle transcripts. I really bring up these experiments because they really set the basis for the methods that we developed later on. So rather than focus on individual genes, which are variable sort of based on the methods being utilized and the platform being used, we realized we had to focus on clusters of genes, which can be commonly observed across data sets. This represents a sort of a dimensional reduction of the data. And this reduced version of the data can then be used for statistical analysis, and this minimizes the effects of variation in individual transcripts. And the other key sort of piece of data that came out of this was that sort of these lower dimensions of the data set really can contain highly meaningful information. For example, transcripts derived from the Y chromosome, this is a simple toy example, but transcripts derived from the Y chromosome are only observable in humans and in tumors from male patients. So to sort of demonstrate a little bit about this dimensionality, we took the transcripts from this original experiment that defined the differential outcomes, and we mapped them onto human osteosarcoma tumor data sets. So the cell cycle transcripts associated with the outcome, they can be observed in the tumor tissue, but they're not in the first few components of the data set. So we're able to see them. They likely have meaning, we realized. And without being able to verbalize this at the time, what we really needed to do was develop a method to systematically analyze transcriptional data using dimensional reduction, find these common transcriptional events in clusters across data sets, and then statistically analyze these reduced values. So we were extremely fortunate to receive a significant amount of funding from the Sobiech Foundation and the Children's Cancer Research Fund, which allowed us to carry out RNA-seq transcriptome analysis of a large number of osteosarcoma tumors from humans and dogs and mice. And as I repeat, I've mentioned this before, but the human and dog tumors are naturally occurring, while the mouse tumors were engineered in the lab by mobilization of an oncogenic transposon, which can activate oncogenes and disrupt tumor suppressors. So we then carried out the dimensional reduction on each of these data sets, and each of these colored boxes represent clusters of genes which could be identified separately in each data set and then can be mapped across the data sets. So while that wasn't the primary component or cluster in these data sets, we were able to identify the cell cycle cluster in each of these, and they are annotated in red and shown down below. And some example members such as Ki67 and thymidine kinase, Aurora kinase, and BUB1 are marked in their locations. So to show that these are associated with poor outcome, we generated a gene cluster summary score. And to do this, to generate this score for each sample, for each cluster, we simply sum all the log transformed mean-centered values in the cluster. So we can then use this value to generate groups of samples with high and low expression values for the sets of genes. And then we can test survival using these summary score groups. So we divide them into high and low groups and then use Kaplan-Meier analysis. So these scores are meaningfully better than using individual transcript values because they do not, first, they do not suffer the curse of high dimensionality. So that in meaningfully replicable results can be generating using modest sample sizes using this, these approaches, because you don't need to test tens of thousands of different transcripts. And I think these work better because the overall transcriptional state of the tumor is being measured. And this is more meaningful than the many individually variable transcript levels. And specific details of this approach are available in our cited publication. So we can use these summary scores to generate groups to analyze outcome by Kaplan-Meier analysis. Using this process, we were able to clearly replicate that high levels of the cell cycle cluster were associated with poor outcomes in both human and with both dog and human tumors. I point out that the x-axis, or the time, is different. It is about a year for dogs, and while it's about 5 years for, it covers a little more than 5 years for the human tumors. So this really shows that the canine tumors are progressing much more rapidly than the human tumors. So then further, we identified that the osteosarcoma tumors show variable levels of transcripts associated with the presence of immune cells in the sequence tissue. These transcriptional clusters, they contain canonical immune cell markers, and they could importantly not be observed in cell lines, which by definition do not contain immune cells, or should not. We further observed that human tissues with poor outcomes were associated with lower levels of these transcripts than tumors with better outcomes, and that the presence of these immune transcripts were higher in tumors that did not metastasize. So we then went back and were able to validate this finding in some of the older osteosarcoma transcriptional data sets. So the Cancer Genome Atlas really is and remains a fantastic resource that I feel is still underutilized. So we were also able to observe that tumors associated with high levels of cell cycle were associated with poor outcomes in a number of the TCGA data sets. And similarly, samples with lower immune transcripts are associated with poor outcomes in many of the other additional data sets. So these results were generated by using a list of genes which were derived from the human osteosarcoma clusters. These patterns can also be independently replicated by going and applying the whole approach or process to the TCGA data sets individually. And I'll show you a few of these in a second, but if anyone is interested in seeing these associations in any of the TCGA data sets, I'd be happy to meet and go over this representation of these data sets. And I'm putting this together as an informatics thesis project. So around the time that we published that work, the TCGA sarcoma data set came out, and we were able to independently validate. Now, these are not osteosarcomas, but are other sarcoma subtypes. So the cell cycle cluster is shown in red, and increased levels of the cell cycle transcript are associated with worse outcomes. And the immune cluster is shown in purple, and lower levels are associated with worse outcomes in this data set. And I should point out that the members of this cluster could be clearly, if you take this list of genes and do enrichment analysis in our osteosarcoma data sets, you will distinctly find the overlapping clusters. Shown here is the TCGA, the skin cancer data set. The purple cluster here is the immune cluster, and the decreased immune transcripts are associated with worse outcomes. And also, here's the kidney and renal cell cancer outcome associations. Here we can see that while there are a number of associations present within this data set with different clusters, the strongest is the association with the cell cycle cluster. So high levels of dividing cells as measured by transcripts associated with Ki67 and other cell cycle transcripts are associated with very poor outcomes relative to cells which aren't dividing as fast. So we really developed a framework for systematically finding meaningful, repeatable outcome associations in transcriptional data. So in parallel work, I became involved in looking at microRNA transcript levels in tumors. MicroRNA are small pieces of RNA that appear to regulate gene expression post-transcriptionally. Using array-based approaches, we identified microRNAs that showed unique expression patterns in sarcomas. Originally, we profiled a set of over 300 sarcoma tissues representing 22 different types and found that the majority of these sarcomas express unique tumor type-specific profiles. To accelerate this research, we made this information publicly available via website. Importantly for this work, when we looked at the osteosarcoma samples, we found a set of microRNA at the 14q32 locus were highly co-expressed, and the tumors and cell lines derived from these tumors showed lower levels of this set of, I think there's 60 microRNA in this cluster. We also showed that MIR1792 cluster microRNA, which are transcriptional targets of MYC and have their own oncogenic functions, are overexpressed in the osteosarcoma samples relative to normal bone tissue. We further observed that decreased levels of the 14q32 microRNA were associated with clear oncogenic functions, and importantly, they showed that the tumors that had the lowest levels of 14q32 were associated with poor outcomes and metastases. We expanded this into the dogs and were able to show that there's an orthologous cluster of microRNA to the human 14q32 that exists in the dog genome, and that decreases in these microRNA were also associated with poor outcomes in osteosarcoma. And in this series of publications, we also showed that decreases in 14q32 were not generally occurring as a result of the copy number decreases at the 14q32. So I was extremely gratified to see that the human 14q32 results were independently validated by an external group shortly after publication. And again, several years later in multiple independent cohorts. So these two different vignettes really lead the main question that I'm going to try to answer today. Are these three different and independent transcriptional patterns, or is the 14q32 expression pattern related to either the cell cycle or immune infiltration patterns? as part of the funding we received, we were able to generate microRNA sequence profiles from a set of human tumors where we also had mRNA profiles defined from the same tumors. So we hypothesized that microRNA transcript levels should be related to the mRNA transcript levels. Or said another way, there are common transcriptional mechanisms which were likely to regulate both mRNA and microRNA transcript levels. So the question we really were interested in determining was, are the 14q32 microRNA related to the cell, run by common transcriptional mechanisms as the cell cycle or immune infiltrates? So to directly observe this interaction, we generated GCS clusters from both the human and microRNA clusters, and then looked directly at the correlation between these GCS cluster values. And the results are shown here. So we used Pearson correlation, and positive correlation is shown in blue, and the closer to 1, the bluer it gets. So as a positive control, we observed that some of our tumors had a strong muscle signature. These same tumors had high levels of microRNA1, which has really been clearly associated with the presence of muscle transcripts. The correlation was very high between this muscle GCS pattern cluster scores and the microRNA1 cluster scores. And this really shows us that it's possible to see these types of associations. We then looked for microRNA that correlated with the cell cycle mRNA cluster. So the 1792 microRNA cluster correlated to the cell cycle transcripts. And we also found a cluster of microRNA which correlated to the immune infiltrate transcriptional GCS pattern. But of importance to this work, the 14q32 microRNA cluster, which is shown on the bottom, did not show strong correlation with the immune or cell cycle cluster. This piece of evidence indicates that the 3 different transcriptional patterns associated with outcome appear independent. This is currently ongoing work, and we will try to validate some of this with the TCGA data set, where microRNA and mRNA is present from common tumor sets. The logic here is somewhat transitive. So in summary of this talk, we show that there are these 3 strong outcome associations: increased cell cycle transcripts associated with poor outcome, decreased immune infiltration associated with poor outcome in osteosarcoma, and decreased 14q32 associated with poor outcome. So I was really surprised after spending several years working on this, when I heard a keynote speaker at CTOS this year say, "I don't believe there are any meaningful prognostic factors in osteosarcoma." In light of our research, this really bothered me, and I began to look at where this thought line could be derived from, because using our methods, these results are quite consistent across different data sets and across osteosarcoma as well. So a quick search of the literature finds a plethora of prognostic association reports in osteosarcoma, but the majority of these lack independent replication. So the OS literature is riddled sort of with the corpses of papers with prognostic claims that do not validate in the genomic era. In a few cases, work has been retracted after concerns were brought up, but in many other cases, these publications exist, and to the naive reader, it is almost impossible to acknowledge what is real from what is overly optimistic. So I'm confident that our prognostic factors will continue to validate, and eventually a useful diagnostic will become available. In the meantime, we're left to ponder what is sort of really happening in osteosarcoma. So in the past, much of the published work in comparative genomics has been focused on identifying similarities between human and canine and murine disease. But there are really profound differences between canine and human osteosarcoma. There are differences in overall incidence, in survival times after tumor formation. There are differences in the age of onset. Osteosarcoma is a pediatric disease in humans, while it is associated with aging in dogs. Thinking about these led us to look for differential driver events in human and canine osteosarcoma. So the classic model at this point for osteosarcoma is that random mutation leads to p53 mutation, and this then leads to genomic chaos leading to tumorigenic proliferation. Here are examples of human and canine osteosarcoma, and show the copy number changes. So p53 is the most commonly mutated oncogene in both human and canine osteosarcoma, and no other recurrent mutations were observed in common between the human and dog OS tumors. And this is consistent with published work, where P53 is clearly mutated and large numbers of copy number changes are present. We looked at mutation loads as well and found common mutations across both of these human and common numbers in both copy number variation as well as mutation count. We also saw because we also wondered whether older canines would show increased mutations or increased copy number changes. You might say that the older canines show a slightly increased mutation numbers, but really they don't appear to be meaningfully different. Similarly with copy number variations, there's really no difference between old and young canines. We also wondered whether summer tumors might be primarily driven by copy number changes, while other tumors might be primarily driven by mutations. In both human and canine osteosarcoma, we see a cone, so we see both mutations as well as copy number changes. They don't seem to predominantly one or the other. So this figure really represents the massive number of copy number changes that are present in the tumors. For normal tissues, this would be essentially flat, but for osteosarcoma, we see on average regions which are strongly amplified and deleted, and we see this similarly across both the human and canine osteosarcoma. So Sleeping Beauty transposon mutagenesis can accelerate OS tumor formation in mice in a mutant P53 background and can cause OS tumor formation in wild-type mice without the copy number changes that are associated with naturally occurring osteosarcoma. Brenda Moriarty, during his graduate work in David Large-Espada's lab, showed this quite elegantly in his 2015 Nature Genetics paper. So we developed a method to identify use linker-mediated PCR to identify tumor formation in Sleeping Beauty screens. And this method has been used in over 30 different screens. When applied to Brandon's data, we identified PTEN, NF2, and ERAS, as well as a whole host of other factors, including the CSF, including genes previously discussed today. But we failed to identify MYC and CDKN2A because they were on the transposon donor chromosome and were excluded from the analysis. In 2016, Alpay Temi and I showed that we could identify fusions in RNA-seq from tumors generated from transposon insertions. These fusions can tag the insertions and then lead to oncogenic changes in transcription. We ran this method on the data set that was generated as part of the Soviet funding. Here are the direct results of that analysis. On the x-axis is a count of the number of recurrent fusions that are present in the data set. On the y-axis is the average count per million of the identified fusions. Additionally, we found P10 and NF2, as well as other genes which were previously described. We also found it was very satisfying to see that we hit MYC and CDKN2A in this analysis, and it really leads to the credence of the method. It's interesting, since seeing this as well as the other, we were able to identify a canine tumor which had a fusion to ERAS. And in conversations at CTOS last year, I learned that at least one of the human target tumors has an ERAS fusion, but these don't appear to be highly recurrent. So I don't think recurrent fusions are as important as copy number changes in promoting OS tumor formation. So we really wondered if we could use the SB fusion data sets to identify copy number changes and identify key osteosarcoma drivers, and whether these would be different between human and canines. What we found, to make a long story short, because I'm running out of time, is that P10 was homozygously deleted in large numbers of the dog tumors, but only a single copy was deleted in about 10% of the human tumors. And these numbers correspond with what was observed by Foundation Medicine in a screen of over 350 tumors. So the question really became, why are both copies of P10 lost at such a high rate in canine osteosarcoma relative to humans? Well, it has to do with the location of P10 in the canine genome. It's out on the end of chromosome 26. And in many cases, the whole end of 26 is lost. Both copies of the whole end of 26 is lost. I'm starting to run out of time, so I'm trying to figure out. So P10 is part of a small 10-gene Sitney block that has been transferred to the distal end of chromosome 26. And this rearrangement goes back 3 or 4 million years to the ancestor of the gray wolf. In all other species, it is internal. And consistent with the canine, we do not see breed-specific bias for loss of PTEN. We see it in all the breeds we've looked at. So PTEN is a tumor suppressor that inhibits PI3 kinase-mediated phosphorylation of AKT, and this drives proliferation. From this, mechanistically, loss of PTEN should be associated with increased cell cycle GCSE scores, and that's exactly what we see when we examine this in the canine tumors. So loss of PTEN have higher cell cycle GCSE scores. Next, we looked at the overall transcript level of PTEN in dog and human tumors. And I was really surprised to see that in dogs which had an intact PTEN, they also had very high levels of PTEN transcript relative to what was observed in human and mouse and in mouse tumors. So I think this variable range of PTEN allows us to directly ask what the role of PTEN is in osteosarcoma. But before we really do this, we need to show that the copy number is in fact related to the overall transcription level of PTEN, and we do that. There's, for the most part, loss of PTEN copy number leads to decrease in transcript level. It does vary from this in some cases where methylation may be imposed, but we don't see any points up in here which would indicate PTEN transcript where it shouldn't be if it's homozygously deleted. So I then directly plotted the PTEN transcript level against the cell cycle score and see the negative correlation as we might expect from the mechanistic knowledge of this. Well, originally as a control, I checked PTEN transcripts against the GCSE levels for immune infiltration. And I was highly surprised to see that high levels of PTEN have high levels of infiltrating cells, while low levels of PTEN have greatly decreased immune cells. I'm really still thinking about how to interpret these results, so I'm interested in any comments you might have. So it appears that PTEN loss may be fundamentally driving cells to replicate, which may also be linked to generating immune-privileged microenvironment. Really, only by integrating data from our comparative approach using OS tissues from different species were we really able to observe this result. So while the dog tumors may be mechanistically different from human tumors, they may be very important for drug development to identify drugs that work selectively on tissues that have lost PTEN, as well as other tumor types where PTEN loss is more common than in osteosarcoma. So our model for OS development in dogs starts with disruption of the P53 signaling pathway due to loss of function mutation, which results in error-prone replication of an unstable genome that in turn results in genetic loss of PTEN and accumulation of additional copy number driver mutations leading to tumor formation. In conclusion, our model provides an elegant structural genomic rationale partially responsible for the increased incidence and accelerated mortality of OS in canines. This in turn suggests that we have the opportunity to reduce the burden of cancer by using genome engineering to migrate the PTEN gene back towards its evolutionary conserved gene neighborhood. And this really starts us down a path where we may be able to engineer cancer risk out of genomes. In future work, we hope to define the roles of other driver copy number changes identified by the SB screens in human osteosarcoma to identify potential personalized treatment opportunities. The work I described today was only possible due to the collaborative sarcoma program at the U of M, which has allowed us to make great strides in understanding the molecular mechanisms of osteosarcoma, as well as other sarcomas. I'd like to thank all of my collaborators on these projects, specifically Jaime Mariano for introducing me to comparative oncology, Subri Subramanian for getting me excited about the microRNA work, and David for all his sage advice. I would also specifically thank Brenda Moriarty, who generated the mouse tumors, as well as the Zach Sobiek Children's Cancer Research Fund and the Karen Wyckoff Rain and Sarcoma Research Fund for the generous support of our work. I thank you for your time and making it through I think this is about the 7th Zoom talk today. So that's what I have.
Thanks a lot, Aaron. Really interesting. So this is open for questions. If I could maybe start with one. So, you suggest that canine osteosarcomas end up getting funneled along a path that involves loss of PTEN because PTEN is in the labile part of the genome in osteosarcoma in that species. I guess I have a couple questions about that. In human, the rare osteosarcomas with biallelic loss of PTEN or activating mutations in PIK3CA, are those associated with the bad outcome and the high cell cycle low immune infiltration that you see?
Yeah, so I think those are great questions, and I think they've been very hampered by the low numbers of tumors that we have to look at. You know, in breast cancer, you can get 1,000 tumors, and you can assemble replicates of 1,000 tumor libraries to do transcriptional profiling and look at outcomes. While efforts have begun to try to do this with most notably the Target project, when we look at the data quality that's generated, a number of those samples are heavily contaminated with ribosomal RNA, which then changes the normalization in such that it's difficult to compare. You're looking at apples to oranges in the RNA-seq transcript profiles across the target data set. So it's my hope that we're going to be able to sort of use the stratification of driver events from the SB screens to go in and define these specific molecular events caused by the specific copy number changes that are recurrent, and that these may lead to targetable therapy in the future. My concerns after making that statement are that when we do this, we're asking, all roads lead to Rome, and something must happen for an osteosarcoma tumor. We're going to have different routes to get there, leading to the same result, such that they're not separately. And that's why I think the P10, the varying levels of P10 tumors in the canine osteosarcomas allow us to see what P10 itself is doing, because there are P10 tumors that have high levels. And I don't know if, when we look at those other factors, whether we're going to be able to identify similar variation.
Other questions?
I have to ask, Aaron, on nice talk. I'm curious, was there any difference in rank ligand or CSF1 expression between your good and bad dog, or for that matter, human groups?
So I believe that CSF1 is expressed in the immune transcript set. Now, that doesn't mean it's definitively coming from the immune cells. It may be used to crosstalk between those immune cells, but I believe it is in the immune cluster itself, and thus associated with outcome. In ongoing work with another collaborator, we're trying to define that crosstalk environment using purified cell populations.
Since osteoblasts make rank ligand, then that might be kind of interesting. Was there a difference in the good and bad groups in that expression, or do you happen to know that?
Yeah, I didn't have time to go into it today, but the osteoblast population is observable in these tumor data sets, just as the immune component is, and it's similar in its so tumors that have excluded all cells exclude osteoblasts and immune cells, but there is some variation in the presence of the osteoblast transcriptional population, and so the osteoblast, anything that is the osteoblast markers are present in there, and I don't know if that completely answers your question, but that's.
Thanks.
Oh, and so Jaime asked, what about reduced P10 expression in human tumors without reduced gene dose? And I assume he means reduced copy number. Well, I think that there's a methylation program that's going on in this tumor that is leading to silencing of P10 in the human tumors.
Right. And do those have a bad outcome?
Well, if you look at the P10 transcriptional patterns, they're very flat. The variation is small, smaller in the human tumors than they are in the canine tumors.
Right.
So I think difficult to define the differences.
Yeah. We'll move on to the third and final presentation, which is by Kelsey Becklin, who is a graduate student working in the lab of Dr. Brandon Moriarty. She obtained her bachelor's degree in biology here at the University of Minnesota Twin Cities. And she's going to tell us about her work.
Let me get this pulled up here. All right, everybody can see that?
Yeah, it looks good.
Excellent, thank you. And thanks, David, for the kind introduction. And I appreciate the chance to tell everybody about a little bit of the work that we're doing here in Brandon Moriarty and Beau Webber's lab. I feel quite lucky that I get to go at this point in the day, because I think that a lot of the talks that were done earlier really kind of help shape what I'm doing here. So I'm going to be talking about our induced pluripotent stem cell modeling system of osteosarcoma development and metastatic disease. So like we've heard today, osteosarcoma is a bone cancer that commonly is arising within the long bones near the growth plate. And just like Lauren showed, osteosarcoma is most common among adolescents, which is really when these our patients are hitting that pubescent growth phase. And another thing that we know about this disease is that when primary disease, so it's not advanced disease, no metastases, we tend to see decent survival curves, which is here in blue, but once metastatic disease is present, the survival rates drop quite dramatically. And another issue which Brenda brought up earlier was that we've really not had any new treatment options for these patients since the advent of modern chemotherapies. And we also know that increasing chemo doses does not have any long-term patient benefits. And that tells us that we need new therapeutics, but that also means that we need new targetable pathways to go for. And we haven't been getting these pathways identified to us through our current model systems. So when we normally model cancers, we're looking at cell lines, engineered mouse models, or patient-derived xenografts and organoids. And although all of these model systems have many positives to their use, there's also a lot of negatives that are occurring, which might be inhibiting our ability to find these new targetable pathways. So my hope is that we're going to be able to use a bottom-up modeling system, which means we're going to be starting with cells that shouldn't be malignant or have too many mutations in them causing disease. And I'm going to add that in so that we can initiate the cancer from the bottom up. And we're working under a pretty simple hypothesis, is that using induced pluripotent stem cells will make a better modeling system to study osteosarcoma. And today I'm going to be talking specifically about our first aim, which is we're going to be looking at how genomic instability impacts osteogenesis, or the formation of bone. So for me to study how genomic instability plays a role in bone development, I need to know a little bit about bone development. So just a quick reminder is that we know that osteogenic differentiation is really tightly regulated. And what we have is that we have changes to gene expression and transcriptional profiles of cells as they mature from MSCs to early stage osteoblasts to finally mature osteoblasts. And that these changes in gene expression profiles is really tightly regulating the cell's ability to differentiate and that and the balance of proliferation. And what we do know about osteosarcoma is that it exhibits osteoblast-like features, but it seems to sustain undifferentiated osteoblasts, meaning that these cells maybe never reach that mature state. And we also know that there are some genetic alterations that most likely occur early on in development, which then kind of poise these cells to having interference of the normal osteogenic process, so of that normal differentiation of these cells into mature osteoblasts and osteocytes. And also that these genetic alterations that probably occur early on also kind of poise the cells to gaining additional mutations as they're developing. And these mutations likely are disrupting that balance that we have between differentiation and proliferation. And within this subset of cells, what we end up seeing is most likely is there's going to be a population of cells that have uncontrolled proliferation, and that our osteosarcoma progenitors most likely arise from those cells and expand to form osteosarcoma. So when I'm making osteosarcoma kind of from the ground up, I needed to choose some genes that I was most interested in targeting. So I've chosen to target two tumor suppressor genes, so RB1 and P53. And what we know is that these genes are often lost in our patients with osteosarcoma, and that when you lose one or both of these genes, you end up getting massive genomic instability, which is a hallmark of our osteosarcoma cells. I have also chosen to overexpress some targeted oncogenes, and these were chosen based on their commonality within osteosarcoma among other cancers, and they're known to drive and really promote malignancy in cells. So to take a step back and give everybody kind of the broad overview of what I do is I take somatic cells, so skin or blood cells from a patient. They're made into induced pluripotent stem cells, which we're able to genetically engineer at various stages of development. And I can differentiate these down our lineage of interest. We can then study these diseases, their phenotypic response, in hopes that someday we're able to identify new targetable pathways, which we can then turn into drugs, which will get us back into a patient. So today I'm going to show a little bit of data from cells that have no genetic alterations, loss of one gene, loss of two genes, or loss of two genes, as well as my overexpression. I'll also be showing data from various stages of differentiation. So this would be my MSCs and then cells that were differentiated down the osteolineage for either 7 or 14 days. And finally, we're going to have some data that I'm showing you from our Anchorage independent growth and calcaneal injection experiments. So when we're differentiating cells, really that's just changing the transcriptional profile of these cells through the signals that they're receiving from the media and then the substrate that they're grown on. And what we see is that when we turn these cells into MSCs and stain them for common MSC markers, is that they look quite, our cells that are genetically altered, here I'm showing P53 negative cells, and then my wild types are here on the left. And what we see is they look very similar, but there does appear to be some very subtle differences in their the staining of these surface markers. What's really interesting for me is when I differentiate these cells down the osteolineage, is that these differences seem to maybe stand out a little bit more. So this is an Alzarian red stain, which binds to calcium and makes this really bright red color, which I'm able to then quantify on a computer software. And what we see is when we lose P53, there does appear to be a slight inhibition of the cell's ability to differentiate into fully mature osteoblasts, which would be indicative of kind of what we see in our osteosarcoma patients. So now that I have my cells, I'm going to run through a couple short experiments. Here's we're looking at a soft agar assay. So this is when I take the cells and instead of them being able to bind to their local surroundings, the cells have to survive in a suspended state. So the bottom layer here is a solid agar, but this middle layer where I plate my cells is semi-solid, so the cells can't bind. And only cells that are capable of growing in that environment will then grow up over time, forming colonies, which I can then stain with crystal violet, which stains my colonies this kind of dark, rich purple. And what you'll see is that when I have no or very few genetic edits, I don't get any colony formation. But as I increase the genetic alterations to these cells, as we start getting colony formation, which we can then quantify and see our differences here. And here I'm showing from day 7 and day 14 osteoblasts, and the trends tend to hold true through both of these differentiation stages. And next I was able to take my cells, so my gene edited cells, I differentiated them down my lineages of interest and I injected them into the ankle joint of mice, and I allowed these tumors to grow up. And I was able to harvest these tumors, and they're out for histology and RNA sequencing currently. Couple things that we found when we injected these cells into animals is that when I injected cells that were only differentiated to the MSC stage, the only time I ever got tumors was in my cells with a double gene knockout and overexpression. But as I differentiated these cells down the osteolineage, what I started to see was that it took fewer genetic alterations to induce these animals into having tumors, and then their survival rates decreased as the genetic alterations increased. We also got a little bit of histology back, and what you're able to see is that between our various stages of differentiation, as our tumors look a little different. At the MSC stage, they tend to be very homogeneous and they kind of look similar, but as these cells differentiated down the osteolineage, as we started to see a bit more variability, and by day 14 osteoblasts, when those were injected, as we started to see quite a few cells that displayed unusual characteristics through histology. And if we compare that to patient-derived samples, which I have here on the bottom, what we see is that the patient-derived samples also have a lot more of that cell variability that we're seeing in our later stage osteoblasts. So for us, that was really encouraging data back, and we're really excited to get the remainder of this experiment done so we can really see if our model system is going to lead us into new understanding of osteosarcoma development and progression. And with that, I just want to thank a few members of my lab, particularly Rebecca Madden, who has taken over quite a bit of the cell work on this project. And she does have a poster out there if anybody would like to look at additional data or look at the data more closely. And we are funded by many different sources, but would just like to thank all of them for their efforts here. With that, I can take some questions.
Thanks so much, Kelsey.
Anybody, so there we go.
Questions for Kelsey?
Kelsey, this is Logan. Sorry, my baby was squealing, but did I hear you say that you get different tumor uptake depending on the stage of the differentiation?
Yeah, so with our MSCs, they only form tumors in the cells that had overexpression of MYC and RAS. But when we went to the osteo lineage, I got about half the animals that were differentiated for 7 days. With the P53RB negative cells, they form tumors. And then by day 14, all of my animals that were double knockout or the overexpression formed tumors.
And I guess my question is there any suggestion in the human tumors that, similar patterns of mutation are deriving in different stages of differentiation, in vivo?
For that, I'm not fully sure. There are a few things that we're wondering, there could be some like culture specific instances that are happening. So when we karyotyped these cells, there does seem to be a pretty big jump in large genetic alterations that occur as these cells differentiate down the osteo lineage, which we don't see in the MSC lineage necessarily. So whether that's something specific to osteo, which would occur in a human as well, that I'm not sure of.
And then secondly, are you getting chromatin profiles at each stage? Because I would love to do the same thing with the osteo data that Beau and you are doing with Ewing's.
Yeah, so for these specifically, I haven't done the ATAC. We have with the Modiano group, there is some preliminary data there with them using ATAC, and we are also setting that up to run through with these cells specifically as well.
Great, thanks.
Any other questions for Kelsey?
So Kelsey, I'm curious how many more cell divisions there are between the MSC stage and the osteoblast stage. If you just like an estimation.
Yeah, so usually when we actually plate those out for an osteoblast differentiation is they get plated fairly heavily. So they should only undergo a few cell divisions, but I haven't quantified that fully yet to know how many exactly that they're undergoing.
Interesting, just because again, if you're saying you're now seeing those large, gigantic pieces of the genome being shuffled around, but there's not a lot of cell division to kind of help that occur.
Yep.
Cool.
All right. If there are no more questions, we'll move on to the end of the conference, which will be closed out by Janelle Calhoun, who is the Executive Director of Rain and Sarcoma and has a few words to say. She is a graduate of St. Olaf College with a major in mediation and dispute resolution with a broad background in industry, including as a pharmaceutical rep before becoming the Executive Director of Rain and Sarcoma. So thank you, Janelle, for joining us.
It is an honor and a pleasure to represent so many amazing families. I'm just going to pull up my slide deck. Emerson, I might need a little help with the camera. Actually, for whatever reason, I'm having an issue.
I'll send you an invite to share.
Start my video. Oh, perfect. Thank you so much. I appreciate it.
Okay, now we can see you.
It is an honor. We'll just get the slide deck up here from the beginning. And 20 years ago, one of the most intelligent, compassionate, hardworking women I know, who I had the honor and pleasure of calling one of my best friends, passed away from synovial sarcoma. And her name was Karen Wyckoff. And her vision, her view, I remember her saying she felt so alone, she felt so incredibly isolated, and she didn't want other patients to feel what she felt. She wanted kids to go to kindergarten, not to go to chemotherapy. And everything that all the researchers are doing, everything that I'm hearing is just, she'd be so proud, she'd be so excited, she would be so pleased. She talked to our fair carousel, and that's how Raynaud's sarcoma gets the whoa, or kind of pull back on the reins to rein in or stop this disease state. And Karen was amazing at bringing families, people, individuals together. She had asked our fair carousel to have a gathering. She was feeling lonely, and 7 people came together, one of which was Barb Inglesby, who was treated at the University of Minnesota. Dr. Katie Dusenberry was Barb's oncologist and radiologist. And Barb is still with us today. Everybody else has since passed away. But with this impetus, with bringing all these families and patients together, that's how Raynaud's sarcoma started. It bubbled up through everyone pulling together to really focus on educating the public and medical community about sarcoma, supporting patients, loved ones, and funding research, which is why we are here today. And a big part of why we're together is, I mean, here's a photograph of, over 25 to 35 sarcoma patients. We get together once in January, a couple other times throughout the year. And the sunflower moves its face to the sun to draw strength and energy. Sunflowers will look to each other to draw energy. And I think the patients and the families of Raynaud's sarcoma very much do the same thing. And I also see a lot of the researchers. You guys work together, you collaborate together, and you draw strength and information. And just hearing the questions going back and forth with digoxin and propranolol, reaching out and talking from Mayo to the University of Minnesota to Children's was really exactly what we were hoping would happen from bringing everybody together today. We are volunteer-led. Blake Hastings is a Ewing Sarcoma patient treated at the University of Minnesota. A number of our board members have either been experienced the loss of a loved one, or are they themselves a survivor of cancer. We also have a number of absolutely fantastic physicians across the board on our board of directors helping guide our mission. We are also led by 6 different major committees: development, patient and family support, red flags, research, finance, PR, and marketing. I am the executive director. I was the first paid staff member. We have now since brought on board a director of development. In addition to just this last week, Katie Ingleby joined. She is our sarcoma education and programs manager. And that's going to be very instrumental. In the slides ahead, we wanted to share some things that is happening with the Red Flags Sarcoma Education Committee. There's a number of tools that the community has that we like to help with faster, more accurate sarcoma diagnoses. When you look at that photograph with the 25 to 35 patients, you will see that so many stories. Oh, it's growing pains. Oh, don't worry about it. It's a cyst. It's just a little fatty something. Don't worry about it. It's such a typical traditional component that so many of our families share. So we've come together and we've developed a number of videos, brochures, tools. I want to highlight in particular an electronic medical records best practice alert. I do believe Dr. Randy Hurley is on the call. He's been in and out. He's an oncologist at Regions Hospital. Over the last 4 years, he's worked incredibly closely with our Sarcoma Scholars program. Back in 2009, a patient who again was seen with Dr. Katie Dusenberry. Her name was
Say, Janelle,
yeah.
sorry to interrupt, but someone just asked if we're supposed to be seeing a presentation.
Actually, we are.
Oh, we're not. I wasn't sure either. And then Alpi asked me.
I put together a nice slide deck for you guys. Oh my goodness.
Oh, I want everyone to get a chance to see it. So
yeah,
somehow it didn't share.
All right, let's see. We've got the video, we've got my voice. The slide deck's pulled up and it's actually like literally on my screen. David, thanks. Oh dear.
There's a button at the bottom of the Zoom field called Share Screen. You have to click on. It's green.
Is it green? Oh my goodness.
Yeah.
Hello?
And then you can select yeah, that's working.
I have a very active desktop.
Yeah, now if you put that in presenter mode, we'll see.
From the beginning. All right, can you see that now?
Yes.
Oh my goodness. I am so glad you said something. That's slightly mortifying, but we'll live, right?
No worries.
So Karen is on the left, our fair carousel is on the right, and that is how we get our logo. And to the left, you can see our logo from back in the day. On the right, we're celebrating our 20th year. And to bring everybody together with this amazing conference is another of our celebratory things to bring forward. Here's our mission statement. Again, instead of just verbalizing out loud, education, patient and family support, and research are the crux of why all of our families are together. And I'm so proud of this photograph. Logan, you might recognize Phyllis Balto in there. We've got just a number of patients and families that are treated between Mayo Clinic, Children's. A number of families travel from the Dakotas, Iowa, and Wisconsin. But the community support is absolutely incredible and outstanding. Our board of directors, here's a great listing. I want to highlight Blake Hastings, who is in fact a Ewing Sarcoma survivor. And I know he is a driver to ensure we are doing the best possible, best-in-class, first-in-class research for all patients across the board. Brain and sarcoma committees, development, patient and family support, red flags, research, finance, PR, and marketing. Again, all these are driven by community members. These are all volunteer-led initiatives and outreach and collaborations to advance the mission of brain and sarcoma. Education, we're doing a number of items. I'll just hit the last bullet point, especially. The community over the last 15 years has put together a patient education notebook. It is handed out at Children's Hospital, University of Minnesota, over at Mayo Clinic, and we've now expanded to Duke University. Brain and sarcoma does have a tendency to expand through relationships. So especially as grad students move and shift and grow, stay connected. Let us know where and how we might be able to offer educational tools and resources to do the most good that we possibly can, because that's ultimately why we're here, to save lives. And about just over 10 years ago, Katie Dusenberry worked with Jan Modelin, who was one of her patients, to become kind of the point person to have sarcoma. We had third-year medical students apply. They get a small stipend, and they go through a pretty intensive boot camp. A number of Mayo Clinic, University of Minnesota, and children's sarcoma specialists will come together for a night, put the students through very rigorous talks of their specialties. And then at that point, the sarcoma scholars educate their peers as they're going through medical school. We've now had 50 program participants. If you guys can see David Sipinski's smiling face, he is our 50th scholar. And right beneath him, Guy Gunther. Guy is helping edit the newest 8th edition of the Patient Education Notebook. So we've got a number of really talented individuals. I want to highlight Mayo Clinic. The very first sarcoma scholar was Brad Stitch. Brad now works as an oncologist at Mayo Clinic, coming through training here at the University of Minnesota. Rainin Sarcoma had always just focused on funding at the University of Minnesota, and there was this incredible young woman by the name of Hallie Ann Brown who had an osteosarcoma at the base of her skull, and she was diagnosed in March 2012. And I don't know why it seems that sarcoma has a tendency to find the best, the brightest, the most energetic, the people with the most amazing capability and future and potential. And that was definitely Hallie. And she suffered, and we've worked closely with Hallie's family. Back in 2016, her mom and her dad worked with some family relatives, and Rainin Sarcoma received a $100,000 gift. It's called from the Barbara Bradley Backguard Family Foundation. And the gift and the promise to Hallie and what we want to do to honor Hallie and her family is, again, broaden the funding locations for sarcoma research, which is how Rainin Sarcoma shifted and started collaborating in research work with Mayo Clinic and also Children's Hospital, which has just been extraordinary. And you can see kind of the rich collaboration, through the past couple hours of today. In addition to that, a diagnostic tool has been developed. Hearing so many people say, I wish would have caught it early. I'd never heard of it. Sarcoma wasn't even in the curriculum at the University of Minnesota School of Medicine until 2009. So there's still a number of primary care professionals that don't know anything about sarcoma, at which point Dr. Randy Hurley stepped in. He started collaborating with a number of our sarcoma scholars, first of which was Dr. Robert Gow, Mel White-Abersham, Andy Hughes, and now just lately Nick Reiner, who is going to be presenting this cumulative work at the CTOS annual meeting in next November. And here's just an image of Nick's poster. I don't know if Dr. Hurley can step in and speak to this at all. I know he's been off and on the call, but Dr. Hurley worked incredibly hard, and watching him over the last 4 to 5 years mentor students, give them experiences, he's just been absolutely outstanding to train the next generation of physicians and scientific minds coming through. This diagnostic tool has now been tested at 5 different Health Partners clinics. We're incredibly pleased to let everybody know that it has caught sarcoma early. And White Bear Clinic, an older gentleman had a lump, and his primary care physician textbook followed everything in the best practice alert. The man saw Dr. Christian Ogilvie at the University of Minnesota. Everything was textbook. They caught it early, proper biopsy, a proper finish. And now I think my understanding is this tool will work to support Dr. Hurley to get it implemented throughout Health Partners. It's Epic-based. We're going to ask for help and support throughout the community to get this tool into more Epic medical systems to faster catch and more accurately diagnose sarcomas. At our different partner facilities, we do, in addition to the patient education notebook, we do a care package that helps patients feel less isolated, feel less alone, let people know that they're in community with others, and we invite them to our annual winter gathering. In summer, we've got a picnic and party in the park. We hope you'll join us. We hope to be in person, but if we're not, we can do another virtual event. You may or may not have joined us last July. And we also do a Rain and Sarcoma Remembers event through patient and family support for grief and loss, because we do know that there is way too many unhappy endings when it comes to sarcoma. If you do have any patients, family, or friends, the second Tuesday morning of the month, we do a community time. Now it has gone virtual, but it builds a lot of connections, it builds a lot of support, and it is professionally moderated by Vicki Strong, who's got a strong background, no pun intended, in leading people through tough times and discussions. We are incredibly pleased and excited and happy. Dr. Brenda Weigel did some number crunching, and through the years with Rain and Sarcoma funding research at our 3 partner groups and on occasion at SARC, we have leveraged the $2 million that's gone on to help improve patient outcomes. It has in turn provided a $13.4 million in leveraged sarcoma research dollars just at the U alone. I remember, Logan, I think the first time I heard Brenda talk about, you might have received one of the first research awards Rain and Sarcoma gave back in 2004. You took that data and you did your magic, you did your number crunchings, you did all the scientific components, you pivoted, you turned that into $1 million grant the next year. And then that's when everybody kind of realized, oh my gosh, it's amazing what we can do if we partner together. And I think that's been a strong part of our culture through the years, is that partnering. And we were able to do it because of Hallie Ann Brown, because of the need for families to come together and work together. We're really proud of the research grants that were awarded this January. Dr. Erin Dickerson, again, more work with propranolol, very excited about that. Dr. Jaime Modiano, you've been prevalent throughout this whole entire event, really nurturing the students in the next generation. And then a new and upcoming phenomenal brain, Dr. Ruping Sun. So we're so honored to see what's going to happen here. And I think 2022, we'll have everybody back in a symposium presenting findings. This year, rhabdomyosarcoma research went to Dr. Fabrice Lucien at Mayo Clinic. That happened at the fall fundraiser earlier this October. And Dr. Chris Ann Schultz, again, building on Dicer-1 related protein work. Really want to call out and highlight Dr. Scott Acuno. I am not sure if he's on or if Dr. Brittany Siontis or other Mayo Clinic representatives are on, but I remember talking to Dr. Scott 8 or 9 years ago because there's so many patients who've come forward and said they might be treated at the University of Minnesota, they might be treated at some other area or event, and they're getting seamless care. Dr. Skubitz, Dr. Dusenberry, Dr. Acuno, like just hearing behind the scenes, because people have been coming together through Rain and Sarcoma, the doctors know each other, the patients feel that they've got great outcomes, there's dialogue going back and forth. And I think through these research symposiums, we're going to only strengthen and build on positive results to only continue improving outcomes for more patients and families. Rain and Sarcoma also has a YouTube channel. There are a number of educational videos, everything from 2 to 3 to 4 minutes to 24, 25, and 26 minutes long. One of our latest is with Dr. Emily Greengard, and she is talking with one of her patients, Serena Morrison. It's a fabulous tool. If you see an area where it can help, please reach out to me, reach out to the office, let us know what we can do to support, assist, help with education, get the right volunteers coming forward. We are looking forward to January 31st. There will be a patient and family winter gathering. Sounds like it's going to be virtual. Please get on patients' calendars as they come through. We will have a Sarcoma Research Golf Classic. We raised just over $40,000 last month, which in turn is going to be funding for the 2021 cycle of research. We'll have a party in the park Monday, July 26th. Vaccine willing, we'll be in person. We'll see what happens. My money is on our researchers. I know how hard everyone's working behind the scenes. And our fall fundraiser should be Thursday, October 7th. And ultimately, Dr. Largaspada, to work with you and team for a 2021 research symposium. We would be excited and honored to do that. We definitely want to say thank you to Todd and Catherine Brown. They are Hallie's mom and dad. Without their consideration, without their kindness, without their thoughtfulness to help more patients and families not experience the loss that they have. They worked with their family at the Barbara Bradley Backard Family Foundation, which established this pivotal gift to help us grow outreach, grow education, and help improve outcomes for more patients and families. We definitely want to thank Dr. Larry Seymour, who has tirelessly chaired the Ewing Sarcoma Research Committee over the last 5 years. He's been a key member of our board of directors, having stepped down just a year and a half ago. Dr. Largaspada, Dr. Weigel, Dr. Spector, thanks to you and the Masonic Cancer Center, we couldn't have had this partnership and this collaboration happening here in the great state of Minnesota. I want to thank each of the researchers, co-investigators or speakers, our graduate school students. But none of this would have been possible. We wouldn't have been all pulled together if it wasn't for Emerson Fuller. So fellow Girl Scout, and she really cares to do an incredible job and bring us all together behind the scenes. And then, of course, our community donors, our patients, and our families. Does anyone have any questions about Ewing Sarcoma, the work that we do? It's been 20 years of collaboration. It's been pretty impressive. And cheers to 20 more. We'll keep up the good work.
Yes, cheers. Any questions for Janelle before we part this afternoon?
Well, no questions. I just wanted to say thank you for Ewing Sarcoma, and it definitely has played a huge part in bringing together this research community and creating a critical mass for these rare cancers. And I'm not a clinician, but I've met more than my share of patients, both through work and people like Phyllis who I happen to get it. So it's wonderful to have a community organization that supports us. It's very motivating. So thank you.
You're here.
Well, we will keep partnering. And again, Dr. Seymour, I know he's on the call with us as well. We just can't thank everybody enough, and we are not done. We will continue.
Well, thanks so much. Thanks to all the speakers and everyone who attended. And like Janelle said, yes, we hope to do this next year and hope that it'll be in person and we can see everybody. That would be nice.
See ya.
Thanks, everyone.
Bye.
Thank you.
Brenda, if you're still on the call, I'm so grateful. So incredibly grateful. Oh my goodness.
Thank you, everyone.
Emerson, fabulous job.
Have a good afternoon.
Take care.